Hello, everyone. Good afternoon. Welcome to the second symposium, The Biology of Galls. Let me introduce myself first. My name is Gabriel Barros da Silva. I am an undergraduate student of biological sciences at the State University of Feira de Santana in Brazil. I am a member of the chapter Iniciação Botânica, a group of young botanists affiliated to the Brazilian Botanical Society. 4 Brazilian universities from 3 Brazilian states are involved in this project, whose aim is to introduce biology students in botany subjects and promote and support scientific events. This is the first event that the chapter is hosting this year, and I would like to invite all of you to follow us on social media, especially on Instagram. I'll put on the screen right after. Because we have exciting news and events upcoming. All right. I would like to thank the invitation trust of the organizing committee in the work of a chapter to be hosting the symposium. And now I invite the president of the symposium, Dr. Bruno Ferreira, from the Federal University of Rio de Janeiro, to welcome our speakers and audience. Thank you all and see you soon.
Hello, Gabriel. Thank you. Good afternoon, Cecidologies. You are welcome to be here in Rio de Janeiro, Brazil, but in an online way. The second symposium on the biology of galls is an event which will organize firstly as a presential event. I hope that soon you may visit Rio in person in a presential face-to-face event. Well, let me tell you a little bit about this event. With the proposal to integrate the scientists from distinct areas and the students on biology of galls, we aim to organize the second symposium on the biology of galls, which would occur presentially here in Rio de Janeiro, inspired by the excellent experience we had in November 2018 in the city of Ouro Preto, Minas Gerais state in Brazil. There we held a small but rich symposium, which we called at the time as Symposium on the Biology of Galls. That was the first. Brazilian and international researchers were invited, and there was also an integration with the students from distinct universities. The first symposium on the biology of galls revealed excellent conditions to discuss the gall systems in distinct perspectives of science, creating an extraordinarily rich experience for all the participants, students and researchers from distinct areas studying galls. Let's remember a little bit on this event now with a video prepared by Professor Dr. José Isaías from UFMG, Federal University of Minas Gerais, and Professor Dr. Hildeberto Souza from Federal University of Ouro Preto. You will see that these scientists are also nice singers. The video shows some moments of the first symposium on the biology of galls. So, Gabriel, let's watch the video. Okay. So this is an amazing place in Brazil, Ouro Preto. And this incredible event in Ouro Preto inspired the realization of the second symposium on the biology of gold. This event, which would occur in the Center of Environmental Studies of the State University of Rio de Janeiro, in the island called Ilha Grande, at the state of Rio de Janeiro in Brazil. Well, the pandemic has arrived, stayed, and is still there. So our challenge was to turn that rich experience in Ouro Preto and repeat that rich experience, but in an online event. This online event counts on with the presence of the most competent scientists which study goals around the world. Around the world. I would like to thank the FAPERJ, the Foundation for Supporting Research of the State of Rio de Janeiro in Brazil, for the financial support for this event. I thank all the organizing committee, Drs. Rosi Isaías, René Carneiro, Valéria Cid Maia, Denis Coelho de Oliveira, Marcelo Guerra, and the mediators, the chapters of the Botanical Initiation Project, with the help in the transmission and the mediation of the roundtables and presentations. I thank all the 14 invited lectors and the authors of the 17 video posters, which will be presented in the 3 days of this event, and more than 170 subscribers for this event. I hope everyone can participate in the chat, making contacts, and that we may have great discussions in this event. Please also subscribe to our channel in YouTube, our channel, No Tropical Goal Group. So to finish, let's see a video of the Ilha Grande, where I hope the event may occur soon in more adequate conditions. The video was also prepared by Professor Rosi Isaías and Professor Hildalberto Caldas. Thank you very much for this video preparation. So guys, let's go to Ilha Grande and let's go discuss goals.
Pois eu quero me aprumar. Vou tratar você com força bruta, pra poder me reabilitar. Pois essa vida não tá sopa. E pergunto com que roupa eu vou pro samba que você me convidou. Com que roupa eu vou pro samba que você me convidou. Agora eu não ando mais fagueiro, pois o dinheiro não é fácil de ganhar. Mesmo eu sendo um cabra trapaceiro, não consigo ter nem pra gastar. Eu já corri de vento. De farrapos, eu vou acabar ficando. Eu já corri de vento em popa, mas agora com que roupa? Com que roupa eu vou pro samba que você me convidou? comigo vai eu vou pro samba que você me convidou, pro samba que você me convidou, pro samba que você me convidou.
So let's discuss goals. Let's begin the event. Thank you, Gabriel, for the introduction.
And now, we will have the opening lecture with Dr. Daniel Burkhardt. And I'd like to invite Dr. René Carneiro from the Federal University of Goiás to introduce and welcome Dr. Daniel.
Hello, can you hear me well? Yeah, it's very good to be here today. I wish everybody a great symposium. It's a pleasure to welcome you all, and even a greater pleasure to welcome to our symposium a person like Dr. Daniel Burkhardt. And I'm gonna talk a little bit about his work, so you can, get to know who he is. But by his presentation, you will see it's much more than that. So Dr. Burkhardt is an entomologist at the Natural History Museum Basel in Switzerland. He has a PhD in natural science. He obtained that title at the ETH Zurich and did a postdoc also in Liverpool in the UK. So Daniel Burkhardt is interested on the classification, phylogeny, biogeography, and biodiversity of insects in general, but particularly on the Psylloidea and the Pelorigidae, which are hemipterans, and as well as Passandridae, which are coleopterans. Other particular interests are on the coevolution of Psylloidea and their host plants. So it's a great pleasure to have Dr. Daniel here with us today, and I hope you enjoy. Thank you, Daniel, for being here with us today.
Thank you very much for this very nice introduction, René. And ladies and gentlemen, I'm very pleased to be able to talk about psyllids, goals, and recent developments in psyllid taxonomy and systematics, which sort of help to improve the picture we have of this group. First, I would like to introduce the psyllids. They are a group of
I think we had a little problem with the connection of Dr. Daniel. We'll be waiting a little bit to see if he will come back.
Do you hear me?
Now we do.
Okay. Should I start again?
Go to the beginning of the slides. You can continue.
Oh, okay. So I was saying that adults, they range from 1 to 10 millimeters, and they're always winged. This is unlike, for instance, aphids or scale insects, which are often apterous. Then they also have simple life cycles. you have a sexual reproduction, adults, the female lays eggs, and then there are 5 instars of immatures. The reproduction is always sexual, unlike, again, aphids, where there are often phases which are sexual and phases which are asexual within the same species. Psyllids are phloem feeders, and phloem is a very unbalanced diet, and to overcome this problem, there are several interesting adaptations. So, for instance, they have a filter system. You see here the intestines of psyllids, and the foregut links via these loops directly to the hindgut. And this is a mechanism to make the excess of water and sugar go directly from fore to hindgut without sort of occupying the midgut where the digestion takes place. Another adaptation are symbionts. Psyllids have a series of bacterial symbionts, and the primary symbiont is Bacterium candidatus Carsonella, and that's a bacterium which has a very small genome. In immatures, these bacteria are concentrated in this bacteriome. Another adaptation is the secretion of honeydew and wax. You see here these wax threads. These wax secretions can be sometimes very spectacular. Here is a species of the genus Hollisiana from Costa Rica. You can see here ants collecting honeydew and these spectacular spiral-shaped waxy secretions. And then another adaptation to this poor diet is the formation of galls. It has been shown in a species from Central Europe, Trichochermes walkeri, which develops on Rhamnus catatica, a small tree. It induces their leaf roll galls. These galls, the leaves which are galled, they stay on the plant for quite a long time, much longer as those which are not galled. And there is another species of psyllid which comes and feeds on that. And it seems that the nutritional value of these of the phloem of these galled leaves is much better than the ones which are not galled. And that's why the species which does not induce the galls, Triosa rhamni, prefers also the leaves which are galled. Okay, I would like to talk briefly about the plants which are involved. Psyllids are usually developing on one or a few related plant species, and we call these plants host species. But adults, they often overwinter on unrelated plants. For instance, in Northern Hemisphere, they develop on deciduous eudicots, for instance, and overwinter on evergreen conifers. These conifers are called overwintering or shelter plants. Adults, they migrate also from overwintering sites to the host plant, for instance, and on these migrations, they tend to stop on also unrelated plants where they may feed or where they just may land. So we call these plants food or casual plants. There was an attempt by some colleagues to call all species on which adult and immatures feed host plants. You could do that, but I think you would lose a very important and interesting pattern of plant psyllid associations by the immatures if you would do that, because then most species would be polyphagous. So we have many species which are monophagous. This is Syllabaxae, a species which is monophagous on box, Buxus sempervirens. And you have many species which are oligophagous, like this species, Glycaspis primulcomiae, which develops on a series of eucalypt species. And then you have a very few, less than a dozen species of genuinely polyphagous species. Here is an example of Bacteristra cockerelli, a species which is associated with Solanaceae, which is a major pest there, but that develops also on other plant families. In addition to this, many related psyllid species, they tend to develop on related plant taxa. So the idea that there is co-speciation between psyllids and their host plants is worthwhile to investigate. Now, I would like to briefly talk about interactions between the psyllids and the plants. You have many psyllid species which develop openly on leaves or plant or twigs of shoots of their host plants. You can see here a species which secretes these waxy wax secretions where you don't see any deformation at all. And then you have other cases where high populations of the psyllid deform the leaves on which they sit. Now, these are probably not genuinely gall. It's sort of a mechanical effect of the leaf rather than a chemical effect. And then you have the proper galls, like here, these beautiful galls on a Psidium species, where the plant reacts on the feeding of the psyllids. In addition to this, there is also another kind of impact of psyllids on the plants. These are the witch's broom. I show here an example of Dinaritaina melanota. This is an African or Southern African species on Syzygium cordatum, a Myrtaceae, and wherever this psyllid feed or the immature of this species here, where they feed on a plant, you see the occurrence of these witch's broom here. So the flower part or the fruit is completely deformed to this witch's broom. In plants where the psyllids is not present, there was never, this witch's broom could never been found. It is suggested that this witch's broom may be induced by a phytoplasma, but at the moment we don't know, and more information, more research will be necessary to look into this problem. I would like to go back in the history of galls. I'm sorry, I have problems to forward. Sorry. There is a problem. Can we get out and in again? I'm very sorry. Now it's working again. Okay.
Did you finish the lecture, or did you have a problem?
No. We have a major problem. I think soon it should be on screen again. Okay. Can you see the screen now?
Yeah, it's on the screen. Can you see it?
It's now good. Okay. Then I continue. I would like to talk about the history of research on galls, and the oldest publication on ciliate galls, and the oldest publication on ciliates in general is by a botanical and anatomical professor who lectured at the university here in Basel, and he published a book he called Theatri Botanici about plant species he knew. That was the botanist Caspar Bauhin, who published this volume in 1620. And he described and figured there carex, the plant without galls, and then here a specimen where the flowers are galled. He didn't know by which organism that was, but he mentioned that there is sort of something funny about these flowers. And here you can see the gall, a photograph of this gall, which is induced by Levia jansai, a species which is developing on Juncus. Just as a reminder, the Marcello Malpighi is considered as the first person, the father of Cecidology. And his works were more or less published half a century later than the works of Bauhin. The morphology of ciliate galls is quite diverse. You have some galls which look like crumpled leaves, and the immatures develop inside these crumples. A very common type of these pit galls on the leaves. You can see here a plant which is heavily galled. Another frequent type are these closed, either globular galls or conical galls. And very often you find also blister galls. That's one on Myrtaceae by Statilia. Then you find also cylindrical galls in some groups and leaf rolls, which are more or less tight and more or less thick. All these galls are on leaves. We have also galls on other plant organs, for instance on stems. That's by a species Calophyia rubra from Chile, which induces these stem galls. They are also popular and they are on the stem. Then you have you have flower galls like the ones I've seen before by Levia jansai on Juncus species. And there are also a few species which induce galls on the roots. Here is an example of a Palearctic species, Craspedolecta subpunctata, which induces these galls on the roots of Epilobium. This is in fact a Holarctic species which occur throughout the Palearctic and in North America. The stage which induces the gall is generally the immature, usually the first instar, but there are also some exceptions where already oviposition and the egg start inducing the gall. Such an example is the gall on Egopodium podagraria. That's an Apiaceae, herbaceous plant, widely distributed in Central Europe. And the Psyllid Diosa flavipennis, you see here adults, and you see immatures. This is the last instar with these wax filament which surround the body, and here the first instar. And here in this case, the gall is induced just by the oviposition when the female sort of slightly opens the skin of the plant leaf and then inserts the pedicel of the egg, and this induces the galls. And then from that, the immature, the first instar, emerges and starts feeding in this gall. I mentioned before that related psyllid species tend to develop on related plant species, and this suggests that psyllid hosts are not homogeneous over the whole plant system. There is a recent paper by David Ouvrard and co-authors. They looked at psyllid host plant and how they are distributed over plant families. And here in this table, you see the most important psyllid host families in terms of associated psyllid genera. And here you see the number of importance in terms of botanical plant species in this particular plant family. So in Fabaceae, that's the most important plant family for psyllids, but this is also the third largest plant family. So in a way, it's not surprising that you have many species there. Second most important psyllid plant host family is Myrtaceae, which is only the 9th largest plant family. Then the third one is the Asteraceae, which is the largest plant family. So again, it's not surprising to find this among the most important psyllid hosts. But then you have several smallish plant families like Rutaceae, Anacardiaceae. You also have Sapindaceae, then you have Moraceae, Malvaceae, Rosaceae, which are considered important plant families. If we look at the most important psyllid hosts, what they represent in terms of species inducing galls, then we can say there are several families which have few gall inducers, like the Fabaceae, Sapindaceae, Malvaceae, Rosaceae. You have others where there are many psyllids on Myrtaceae, they induce also galls, or on Rutaceae, on Anacardiaceae. Here you see some quite spectacular galls on fig. This is the edible fig, a species from the Near East, where this Porosilla species is considered to be a pest of figs. I would like to look at these host patterns now relative to the phylogeny of psyllids. And I show you here a recent phylogeny which was based on about 400 psyllid species. That means it's 1/10 of all known psyllid species described worldwide. The colors, they reflect different psyllid families. You see here a reduced family cladogram as it is, or it will be published in a paper very soon, where the classification of psyllids is likely remodeled. We have 7 families, here the Aphalauridae, and here on top the Triosidae. The size of these triangles reflects the species which were used in this analysis, and they do not represent the true size of the family in terms of species included in this family. Now, looking at these different psyllid clades, I would like to point out some which are restricted to particular host families. So, for instance, Rhinocolini are mostly on Sapindales. The Phacopteronini are also mostly on Sapindales, and also Apocinaceae. Spondyliaspidaini are generally on Myrtaceae. Homotomidae are exclusively on Moraceae. Pachycelaini are exclusively on the genus Celtis. Sidarini on Malvaceae. This small family Mastidrimatidae on Meliaceae. And then many Colophaids are on Sapindales. Acaciaini on Fabaceae. Macrococini on Fabaceae, mostly. Syriacromini again mostly on Fabaceae. And Silini, this Aretina group, this clade, is exclusively on Fabaceae. So in general, any species of this family Psyllidae is associated with Fabaceae, but Psyllidae is the largest family with about 1,500 described species, and there are also many other unrelated plant taxa. Now, if we look at these host groups, we see that Sapindales occurs in several families and is relatively to the size of the plant families is overrepresented among these psyllid hosts. If we look at gall inducers among these clades, so there are some, for instance, this Ozotriosa, Schizotriosa complex is gall forming, again, Colophaiini, Pachycelaini, and Phacopteronini. Others are mixed, and there are some where there are no gall inducers at all. So if you look at this pattern globally, it sort of seems there are many groups of psyllids which are polyphagous and not really interesting. If you look in detail at a particular subfamily, for instance, here, the Aphalauridae, you see here this clade in the cladogram, and here a cladogram of the genera of this subfamily. This is a mostly north temperate subfamily. And you see that, again, the different genera within this subfamily, they are more or less restricted to a family, sometimes even to a genus. I would like to concentrate on this small genus here. That's the genus Limataphalara. Here you see one species from Florida, which is absolutely spectacularly colored and very shiny. There are relatively small species about 3 to 3 millimeters, and here an immature. And all these species, as far as known, they induce these leaf rolls. There are 4 described species in Florida, in Costa Rica, and 2 in Brazil. There are another 4 undescribed species in Brazil. And all these species, they develop on different plant species, either or. Here, this Holysea and this species 1 are geographically disjunct. So what we can see here, these species are basically monophagous, and this the diversity in this group can be mostly explained by geographic and host plant vicariance. There doesn't seem to be any close pattern between the phylogenies of the psyllids and of the host plant, so there is no genuine co-speciation. Another example where you have quite the opposite condition are these 2 plant species from Brazil, Copaifera lansdorfi and Hymenea baril, which host a large amount of species. And you have some species which are call-inducers, like these Colophorina species, here an adult of Colophorina, and Orsilopsis, here the calls of Acilopsis. And then you have free-living species, like here these Mitracilla species or the Platycorypha species. In this, in the case of Copaifera lansdorfi, if you look at the distribution of these species, you have here, for instance, quite a series of species which occur in Paraná. And in fact, very often you have 4 or more species on the same tree. So in this case, you could call this tree a superhost, because you have together all these species. In the situation in Hymenea baril is quite different. There are usually 1 or at most 2 species together on the same tree. So you have quite a bit of geographical vicariance between these species, and you couldn't call that a superhost. The literature on psyllid galls is quite extensive in the 19th and early 20th century. Usually these description, this literature is descriptive and often lacks good identifications of the psyllids and of the plants. In some cases, there are several authors which were quite productive in terms of psyllid galls. For instance, Dr. van Leuven, Kiefer, Rübsaun, or Tavares. Fortunately, this very extensive literature was summarized by UERJ in the series of about 10 volumes of psyllid galls around the world. Even though many of these records are still dubious and difficult to interpret. I will talk about 2 or 3 of these problematical cases now. So there is this genus Ozotriosa. Here you see the original description. So there is an explanation of the name, and the description in French says that this is like the genus Triosa, but it differs in the thickened antennal club or antennal segment. This genus was described from India. The description is not diagnostic, and also included 2 species, but did not designate a type species. One species he collected, or he described from Schmidia sikimensis, and the other one from, he said, Styrax. In addition to these problems, there is no type material. Kiefer was in a monastery where they were not allowed to have worldly possessions, so after studying the material, he threw it away. The descriptions of these 2 taxa, one also Triosa lauriniarum, is not, and is not diagnostic, and it could in fact any sort of triosid like psyllid. The other species, it's slightly better, where there is some information which could be used. The names have not been used since 1905, but they have been used in the 20th century, so we have to take them into consideration. In 2018, I published a checklist of the Indian psyllids. I published it together with 2 Indian specialists, and we designated this Ozotriosa styraciarum as the type species of the genus. This, according to the description, it looks that this Ozotriosa styraciarum could be identical or similar to Triosa jambolane, which is an Asian species associated with different Syzygium species. It induces also the galls similar to the ones described by Kiefer. See here these galls, and here they have been cut open. Here is the adult. And you see that the wing shape is sort of similar. So to conclude this, Ozotriosa can be used for a group of Indo-Australian Triosa and Schizotriosa species inducing galls on Myrtaceae. Another example is even more complicated. It's species of Neotriosa and Nototriosa. Kiefer described in the same paper as before the new genus Neotriosa for the species maculae, a species on Maculus scamblii. So again, we don't know exactly what this Neotriosa maculae is, but there are some indications suggesting which group of psyllids it could be. And then Tavares described some galls without naming them from Malpighiaceae, from Bahia, and Crawford studied the material collected by Tavares and described it as Neotriosa tavaresi. Here you see the figure of the galls by Tavares and the forewing of this Neotriosa tavaresi as illustrated by Crawford. And you see here the forewing of Neotriosa maculae, which is quite distinct in many characters. So the assignment by Crawford of Neotriosa tavaresi to this genus was with doubts. Much later, in 2003, Butignol and Pedrosa Macedo reported Neotriosa tavaresi from Paraná from galls on Cydium cattleyanum. And you see here similar galls on other Cydium species. Then in a paper with René Carneiro, we erected the genus Nototriosa for this species by Pignol, which in fact we considered as a new species, another undescribed species, and this Nototriosa tavaresi, which was doubtfully reported from Alp-EAC. Recent fieldwork suggests that there are several additional undescribed species Oncidium of this genus Nototriosa, inducing the same type of, or a similar type of globular leaf galls. Looking again at this illustration of this photograph of by Tavares of this species he described, it could well be that this is a misidentified Cidium species, which would of course fit very nicely into the pattern of the other species. And the fate of Neotriosa was again that until the early 21st century, it was a genus which was not further considered. And Lee transferred some species which develop on Moraceae, on Ficus, to this genus. His reasons are dubious, and it was not accepted. A little bit earlier, Yang described a Rhinocilla shuilensis from Taiwan, which induces galls on Maculos japonica. And Lee described another Rhinocilla maculae from Maculos. Then in a revision of the genus Bactericera, I synonymized this genus Rhinocilla, which is in fact North American genus, with the genus Bactericera. These two species, Schulensis and Maculae, don't belong to Bactericera, and we transferred them back to Triosa. Then in the following, Drojowska erected a new genus for these species, several species which have sort of short or no genal processes. Included was also this Rhinocilla schuilensis. And finally, in a revision of Taiwanese species, we synonymized Neorhinocilla with Triseti triosa, another Asian genus, and suggested that Rhinocilla schuilensis and maculae are closely related to Triosa maculae, and that Neotriosa is a valid genus with 3 included species. You can see here the galls on Maculos, which fit very well the description of galls by Kiefer. Now, these problems are fortunately not too often, and the main work we have to do is describe just many new species in groups which are well defined. Here is an example of a group of triosid species, the Triosa purpuridis group, from which we know 12 species from Argentina and Chile, all specific on purpurис, and they induce all different types of galls. It's usually closed leaf galls or leaf roll galls. Recent fieldwork in Chile and in Brazil produced many more undescribed species, so there are at least 20 undescribed species from Chile, and there is 1 species from southern Brazil, which you can see here the galls of. So all these species are well defined, particularly by the male terminalia, which you can see here. The group appears to be monophyletic and probably constitutes a new genus. Another example of current research is a group of 3 undescribed species, undescribed genera associated with Proteaceae. There's one genus from Africa which is associated with Protea and related genera, and one it's at the moment called Procephala picarinata, induces these very conspicuous leaf galls. These 2 species which are described, they have been described in the genus Procephala, which is completely unrelated. And these, plus another 12 undescribed species, will need the erection of a new genus. Then there is a related genus to that in Central and South America, Rubella, with at least 4 undescribed species. So far, we couldn't find any calling species in this genus. And there is a 3rd undescribed species with 1 described species misplaced in Gyroxilla on an unknown host. We don't know the host there. And seeing that it belongs to this relationship, we could suggest that it may develop on a Proteaceae. These 3 genera are probably closely related, but the group as such is difficult to place currently within the phylogeny of Psyllidae. To conclude, I would like to mention again that phylogenies are very useful to do all sorts of predictions, such as host groups and galling patterns. Psyllids are, to a certain extent, host-specific also at higher taxonomic level. And it appears, or it's likely, that the primitive host association is with Sapindalis. The distribution of gall inducers across Psylloidea is quite uneven, but these patterns are also slightly blurred by the misidentification of many host plants and particularly old records. And revisions are definitely necessary at that level, that all these old records are confirmed with new material. So targeted fieldwork is very important and much needed. And there is a lot of taxonomic and phylogenetic work waiting until we sort of start understanding these very interesting patterns of psyllids, galls, and host plants. And with this nice gall of Neolithus fasciatus, I would like to thank you for your attention. And now, if you have any question, I'm happy to answer them.
Hello.
Hello.
Yeah, that was great. We have a lot of questions on the chat. I told you I would put them on the screen, but I'll read it anyways. Okay. Let me insert the first question. It's on top. There are a lot of questions.
Well, I hope we still have some time.
Okay. Okay, the first question by Rene Carneiro. Daniel, you said the psyllids migrate and overwinter in different species. How far can they fly? Would gall-inducing psylloidea also be capable of migrating or reaching further host populations?
Okay. Usually the active flight of psyllids is not very directed. So in Central Europe, for instance, they migrate between host populations, which are in the lowlands. The lowland here is around 300 to 500, 600 meters above sea level. They migrate to overwintering places. That's the pre-alpine conifer belt, which is at around 1,200, 1,500 meters above sea levels. These distances are sort of in kilometers, tens of kilometers. So I think they can migrate there and back using these convection winds. So that's one thing. Psyllids can also be blown over oceans and over large distances, but this is not active flight. And there, if they are blown in a new area, if the host is there, they can possibly get a foothold and start developing, but usually there is no host there, so they would die. I hope this answered at least part of your question.
Okay, I will invite René Carneiro to introduce Dalva.
Yes, you have answered my question. Thank you. I'm just going to make a small parenthesis here because I was talking to Dalva, and I invited her if she wants to stay online with you, so you can answer questions together as you work together largely on the psyllids. So Dalva, thank you for being here with us today. Dalva will be properly introduced later, but I can tell you that Dr. Dalva Queiroz is a researcher on psyllids in Brazil, and she works at Embrapa in Paraná. And I mean, please be comfortable to share the talk with Daniel. Okay. Thank you, Daniel.
Thank you very much for inviting me for this very nice symposium. I'm very, very happy.
Thank you.
All right, moving on to the next question. Denis Oliveira asks, hi, Daniel. What are the most common places for psyllids overwinter? Soil, host plants?
Okay. The psyllids, it's the adults which overwinter on these overwintering plants. So they're the ones which migrate. They stay on the branches, and they hide there in crevices of the twigs, and they become active and start feeding whenever the temperature is warm enough. It can be that the general temperature is below freezing point, but if there is sun locally, it can be sort of 5 or 7 degrees, which is already enough for the psyllid so that they can feed. Now, there are many species of psyllids which in fact overwinter as egg on the plant or as immature also on a plant, usually in slightly hidden. And there are also some which overwinter as immatures in the galls on the roots. So I think that's about the range. There are some where adults overwinter in the leaf litter. This is known from some willow feeding species in the northern hemisphere.
All right.
So I think that's about variety, the range.
Next question. Christian Alfonso Villagra Gil. In South America, we often see coevolutionary patterns between psyllids, gallers, and native host plants lineage such as Baccharis asteraceae or Schinus anacardiacea.
Okay, I did some research on Schinus feeding psyllids and Baccharis feeding psyllids, and in the case of the Schinus psyllids, there was one problem, that the identity of these Schinus species was problematical. So depending, let's say 15 years ago, depending if you lived in Argentina or in Brazil, the same species had a different name, which doesn't really help to see patterns. Now we have phylogeny for the Schinus species, and we have sort of a bit of phylogenetic information for two groups of psyllid species. And if you compare these phylogenies, there is no real coevolution or co-speciation between the two groups. So
I'm sorry, Daniel, but the question had a continuation. It's on the screen if you can read. I'm sorry for that, but you can go on. Sorry.
Okay. And the thing is that you have some species which are monophagous and others which are oligophagous, which are, there are some Calophyia species. There is one called Mammifex, which occurs in Chile, which develops on at least three species of Schinus. They are all inducing galls, and there are others such as Calophyia rubra, which induces these stem galls, which is monophagous only on Schinus. I forgot the name. Doesn't matter. On one particular Schinus species.
All right. Next question. Question from Valera Maia. Are the galls of Craspedolepta, I'm sorry, I can't read that.
Craspedolepta on Epilobium.
Induced on area roots?
No, they are not. They are in the ground. So the eggs are laid on these Epilobium plants, and the first instar migrates down in the roots, in the soil. And there they induce these galls, they overwinter, and then the second, third sort of instars are down in the ground, and the fourth moves up, and you find the fourth and fifth instar above ground again on the leaves.
All right. Lubia Maria Guedes Garcia asks, some species of Schinus anacardiacea are native to Chile, such as Schinus polygama and Schinus patagonicus. Are the gully psyllids associated with Schinus species also native insects?
I would say they are all native, except for Calophyia schini. Calophyia schini is on Schinus moye, and there it's, I don't know exactly where this originates from. Some say from Peru somewhere. I don't know. But I would say that otherwise they're all native.
All right. Valera Sidmaia. How many psyllid galls are in the Neotropical region and in Brazil?
This is very difficult to estimate. In Brazil, there were, Tal Van Dij did this catalog in 2012 where we reported, I don't remember exactly, around 70 species. Okay, and now we know 130 something species. We estimate that there are about 1,000 species in Brazil alone. And we estimate that we collected in the last 12 years around 450 species. To me, it seems that perhaps at least a third of the species is gall-inducing, a third is not, and a third we don't know.
Right.
There are, as I tried to show before, there are some groups of psyllids or some groups of host plants which predominantly have galls or are gall-inducing. But I think at the moment we know too little to sort of say anything.
All right, the next question is by Valeria Sidmaya again. Do psyllid galls have a predominant shape?
Well, I was sort of trying to show some, and I would say that pit galls is something which is quite common. Also, these leaf roll galls are quite common. But then it depends a little bit on the region, on the geographical region, on the host group. You will find more or less of the other ones. I think in Mutaceae, for instance, these blister galls or leaf fold galls are quite frequent, but you have also many globular gall forms there. So I don't think there is a predominant shape. Alva, I don't know if you have a better answer to this question.
I don't know, probably if you look for specific hosts like Chinus, we have more pit galls, but we also have globular galls on Chinus. And also Myrtaceae that we have lots of galls, but then we have these leaf roll galls, we have globular galls and pit galls. Yeah, it's not, I don't think we have a predominant shape.
All right, the next question by K. McCormack, I guess. Hi, Daniel. Is there a selective hypothesis of gall formation in psyllid? Are any parasitoids associated with gall-forming psyllids, and do they differ to parasitoids attacking non-galling psyllids?
I think I have to answer that step by step. There is no selective hypothesis at the moment. I think we don't really know enough about these patterns. And I think there are some galls which are associated with parasitoids, but I don't see that there are sort of clear differences between parasitoids attacking galling and non-galling psyllids. I think it's more particular psyllid groups are associated with particular parasitoid groups.
Can I say something?
Please.
I don't know if this is the question, but have said if do they differ to parasitoid attacking non-galling psyllid? Sometimes we see that if the psyllids are parasitoid, the color of the galls change, like to reddish or dark. Most of the time they are the same.
Yeah. Okay.
We have one more question by now. I asked them the chat if someone has other questions. Let's wait a little bit.
Okay.
But the next question is Vitor Martini asking, this overwinter gall insect feeds during winter without formal gall?
I think all the cases where adult psyllids overwinter, of course, we don't have galls because adults do not induce galls. The only thing where I can think is species which overwinter as immatures in the galls, and there I would suspect that the microclimate in the gall is temperate, so basically they would feed.
Okay, we have one more question. It's on the screen.
Yes. I don't think so. I don't think that gall inducers are necessarily univoltine. I think very often they are, but sometimes they are not.
All right, Dr. Daniel, we have a lot of compliments to your speak.
Thank you very much. I always like that.
I'll put them on screen or just to use see the comments. Everyone just like there are to Dalva also.
Yes.
Let's wait a little bit if someone asks another question. Isaías, thanking you.
Yes.
Let me see again if there are more questions.
Are there?
Okay, I'll put Bruno Garcia Ferreira on the screen again.
Thank you very much, Daniel, for this amazing presentation on golden psyllids. It's always a time to learn more about the gold industry because they are very diverse. And Dalva, I also like to thank you. And at the third 3.5, we will have the session 1 with Dalva and other lecturers, and which will be about gold inducers. Okay, at the 3.5. May we try to play the videos again because the sound was not okay? Maybe, Gabriel. Thank you, Daniel and Dalva. Do you want to comment something more?
Well, I would like to thank you to have been invited. This is really an honor for me. And it's, of course, Dalva who did a lot also of the brain work and the aesthetics of the presentation. So I think the thanks go also to her.
Thank you. Bruno, I think I can talk more later in my presentation.
Okay. Thank you for being here. And we'll see Dalva in the next session. Thank you, Daniel and Dalva. And Gabriel, let's try to play the videos.
I don't know what to do.
Are we?
No, we're still on.
We are still on. No. No.
chefe da folia pelo telefone manda me avisar que com alegria não se questione para se brincar. Chefe da polícia pelo telefone manda avisar que na Carioca tem uma roleta para se jogar.
Ai, ai, vamos para trás, ó rapaz. Ai, 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 fica triste, seus capazes e verás. Ai, ai, deixa as mágoas para trás, ó rapaz. Ai, 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 fica triste, seus capazes e verás.
Quando apanhes pra não tornar a fazer isso, tirar amores dos outros e fazer o seu feitiço. Olha a rolinha, senhor, senhor, se embaraçou, senhor, senhor, caiu no ar, senhor, senhor do nosso amor.
Senhor, senhor,
Pois esse samba
senhor, senhor,
é de arrepiar,
senhor, senhor,
põe perna bamba,
senhor,
faz gozar. O peru me disse que o morcego visse não fazer tolice, que então saísse dessa esquisitice de disse não disse. Quem for bom de gosto, mostre-se disposto, não procure encosto. Tenho riso posto, faça alegre rosto, nada de desgosto.
ai, 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 deixa as mágoas para trás, ó rapaz. Ai, 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 fica triste, seja capaz e verás. Ai, 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 dança o samba com calor, meu amor. Ai, 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 pois quem dança não tem dor nem calor.
Nice. I think that now everyone can hear the video. Now, the video of Ilha Grande, Rio de Janeiro.
Agora vou mudar minha conduta, eu vou pra luta, pois eu quero me aprumar. Vou tratar você com força bruta, a poder me reabilitar, pois essa vida não tá sopa. E pergunto com que roupa eu vou pro samba que você me convidou. Com que roupa eu vou pro samba que você me convidou. Agora eu não ando mais fagueiro, pois o dinheiro não é fácil de ganhar. Mesmo eu sendo um cabra trapaceiro, não consigo ter nem pra gastar. Eu já corri de vento em popa, mas agora com que roupa eu vou pro samba que você me convidou? Com que roupa eu vou pro samba que você me convidou? Eu hoje tô pulando como um sapo pra ver se escapo dessa praga de urubu. Já estou coberto de farrapos, eu vou acabar ficando nu. Eu já corri de vento em popa, mas agora com que roupa eu vou pro samba que você me convidou? Com que roupa eu vou pro samba que você me convidou? Pro samba que você me convidou? Pro samba que você me convidou.
Thank you very much to Dr. Aziz Ayas and Dr. Hildeberto Caldas for the song and the video, amazing videos. So, Gabriel, can we now to go to a break?
Yes, we are going to a break, a 30 minutes break. We will be back at 3:30 p.m. The live will be on, but there will be a chronometer on the screen, all right? And in 30 minutes, we'll be back. So see you soon. And that's it. Let me put the screen.
Let's chat in the YouTube.
Good afternoon, everyone. Let's welcome to the first session of the symposium. The topic of the session will be Gall inducers. And before we begin, let me introduce myself first. My name is João Victor Nunes, and I'm a biological science undergraduate student at the State University of Feira de Santana, Bahia, Brazil. Today we are going to have 4 presentations with 4 different speakers, and I'm gonna introduce each one of them. The first one is Dr. Dalva Luiz Queiroz. She is a researcher at the Embrapa Florestas in Colombo, Paraná, Brazil. She has a PhD in entomology obtained at the Federal University of Paraná, Brazil, and a postdoc by the Curtin University in Australia. Dr. Queiroz is an expert in psychology with focus on their biodiversity and pest control. The second one is Dr. Valéria Cid Maia. She is a professor at the National Museum of the Federal University of Rio de Janeiro. She has a PhD in zoology by the National Museum of the Federal University of Rio de Janeiro and a postdoc by the Federal University of Minas Gerais. And she is an expert in dipterology with focus on the biology and taxonomy of Cecidology, the most diverse family of gall inducers. And third, we have Dr. Adriano Cavalieri. He is a professor at the Federal University of Rio Grande do Sul, Brazil. She has a PhD in animal biology by the Federal University of Rio Grande do Sul, Brazil. Dr. Cavalieri is an expert in insect ecology and taxonomy with emphasis on the Tisanoptera. And the last speaker is Dr. Gilson Moreira. He's a professor at the Federal University of Rio Grande do Sul, Brazil. He has a PhD in entomology by the Cornell University in USA and a postdoc by the Harvard University, also in USA. Dr. Gilson is an expert on the biology and diversity of Microlepidoptera. And first, we are going to have Dr. Dalva and Dr. Valeria's presentations, and then I'm going to read the questions and comments of the audience for them to answer. And after that, we are going to do the same with Dr. Adriano and Dr. Gilson's presentation.
Dear colleagues, this is a pleasure to be here with this nice group and selected group on goals. I'd like to talk to you about psyllid scholar as a plant pest. Why psyllid can be a pest? Because they feed on the plants, they weaken the host, they damage flowers, fruits, and leaves. They secrete a big amount of honey tea, that it's a very rich substrate for soot mold, and the soot mold is spread over the leaves, and then the photosynthesis is very low. They inject toxin in their saliva, and this can make damage, distortion, and others in the plant. The big problem is some psyllid transmit bacterial disease that can make lots of problems, including kill the plants. They induce galls in some plants also. As an example, as a psyllid is a very big pest, we can cite Jephorina citri, that it's a pest on citrus in several countries in the world, and they are very problematic to control, and that they reduce the production, and also they can kill the plants. We have a group of Cacochylia species that also transmit the phytoplasma for pears and apple. In eucalypts, we have several Spondyloaspididae and Triosa that make damage. And in ornamental and veg fruits and berry, also some psyllids that cause damage. We can say we have few major pests, but many minor pests with local or temporary occurrence. The identification of psyllids often are very difficult for non-specialists. The problem is the absence of a taxonomic base. For example, in Brazil, we have most of our species are undescribed, are unknown. Adding to this, we have many misidentifications. The literature is full, and we have many species that are morphological very similar. We have mate tea in south of Brazil, Ilex paraguariense, that hosts Gyropsila spegazinianas. This psyllid Gyropsila induce leaf blister cause. This is originating from South America, and this is a very problematic pest on south of Brazil. The adult lay the eggs. The adults lay the eggs on the small leaves, and then the leaves start to distort and become just a gall. And then they matures, grows inside these galls, and also the galls grows a bit. And then just when they are about to emerge adult, the galls open and the 15 immature leaves the galls, and then the immatures, the adults emerge. The problem also is Ilex paraguariense is used as tea, as pharmaceutical, and also for cosmetic. And we don't know if this psyllid, with these lots of change on the plant morphology, if they also change the chemicals composed of the leaves. And as this is used as tea, we also don't know if they change the flavor of the tea. Another psyllids we have in Brazil is Triozoida limpata that make leaf row galls on guava. This psyllid is originated probably from Central America, but now it's spread in tropical America, including Brazil. And they make leaf row galls on guava, and this is very problematic in many states in Brazil that the guava is growing for fruit, for juice, and others industrial purposes. And also, we did a potential distribution of this psyllid under the global climate changes conditions, and we saw that it also can develop very well on other countries like India that grows guava, and this guava, it's very important fruit in this country. Triosa tabebuia is a species that uses as a host different species of trumpet tree, genus Androanthus, that are used for ornamental and shadow on the cities. And this Triosa tabebuia induce leaf row galls basically on plants of different ages, but it's more important with small plants on seedlings when they attack, and they can distort the whole plant, and then this plant doesn't grow, We don't have a good plant, and for our ornamental purpose, it's not beautiful. And then in some case, the leaves are also dropped. We have in avocado 4 different species of psyllids reported from different countries, and all of them make galls, different types of galls, like leaf roll galls by Charles Codewey, like pit galls or conical galls by Triosa percy, and Triosa anceps, that they mature start in a pit galls on the underside of the leaves, and then when it grows, the immature grows, these galls become conicus on the upper side of the leaves. In mango, we have a very interesting type of galls. This Apsilla cistellata, the female lay the eggs on the new shoots on the primords of the leaves start, and then these leaves become together, making like a chambers, different chambers, many chambers, and these psyllids are inside these chambers. And in this case, we have these new shoots transformed in galls, and the production of flowers is very low. We have Laurel nobilis, laurel, that it's a plant used for spice, and Laurel triosa alarx induce leaf blister galls. This species native from Mediterranean and introduced in Central and North Europe and America. And they cause lots of distortion and death of the new shoots. The plants, it's like a mess with galls, like roll galls, blister galls, and all together, and then the death of the new shoots, and it become dry, and the whole plant are messy. We have many different myrtaceae in Brazil, and many of them host different psyllids. We can cite as an example Myrciaria dubia, Myrciantes fungi, and Psidium cataliano. All of these 3 species are used for fruit, and in some cases they are also used in street for arborization or in parks for shadow or ornamental. And all these trees host psyllid that makes kind of galls. In the case of Myrciaria dubia and Myrciantes fungens, the galls are type blister galls. And in the case of Psidium cattleyana with Nototriosa cattleyana, that makes globally galls. In eucalyptus, we have in Australia more than 300 species of psyllidia, and 71% of these psyllidia are on eucalyptus. Between these psyllidia, we have 2 families that are important on eucalyptus. Most of them are free living on the shoots or on the leaves, or they live under the layers. But we have also some with psyllids that make galls, with some species that make galls. And on Triosidae, we have a genus Scedotriosa with 12 species that all of them make galls. And then here you can see adult female, and here many leaves with galls. And here we have this layer-like shell where the immature lives under the layers. I'd like to talk about Crucianus latipenis, one species that was described in 1989. The genus Crucianus is original from Malaysia, with 2 species described from Borneo, 2 species from Sulawesi, and 1 species from Ceram. And they have as a host the genus Spongia, Anacardiaceae. And this genus Spongia comprises 12 species. One of these species is Spongias dulcis, that is widely cultivated, and in Brazil it's known by cajamanga. This species probably originate from Polynesia. Crucianus latipenis is a species that was described by Burckhardt and Lauterer, and in 1989. And after that, we didn't find any more citation about this species. It was introduced in Vietnam, Taiwan, Borneo. It was cited when it was described, and it was introduced in French Guiana last year. As this is the first report of these Crucianus latipenis in South America, we are making a publication with this report. And when we almost finish this publication, we have the report that is also in Suriname. This makes lots of distortion, crumpled leaf, raw leaf galls, and the plant is like a very messy. Well, in conclusion, we can say that Psyllidae, we have few major pests that are galling on cultivated plants, but we have many minor pests with local or temporary occurrence, like we can cite this Gyropsila spegaziliana on mate and mate tea. We can say also that galling may cause considerable problems. You saw different types of damage, different types of galls on cultivated plants. And then we should monitor and control these ones that are pests. We need also more field work to understand these species and realize what their hosts are, if they are monophagous or polyphagous. And we also are every day new pests and new introductions. And then we need to pay attention. I'd like to thank you, Rose and her team, for inviting me for this symposium. And I would like to thank everybody for your attention. Thank you very much.
Thank you, Dalva. It's a great presentation. Now let's move on to the Valeria presentation.
Good afternoon. I'm Valeria Cid Maia, meteorologist at Museu Nacional. I'm going to talk about Cecidology, state of art in Brazil. Cecidology is a cosmopolitan family of Diptera with more than 6,500 described species. Most of them are gall-inducers. Due to this habit, Cecidology are known as gall midges. In fact, Cecidology are the most diverse taxon of gall-inducing insects. They are divided into 6 subfamilies, which vary in species richness and geographic distribution. The basal subfamilies are little known and little speciose. They include only fungivorous species. Catophycine with 8 species, Leishmaniine with 102, and Poicondyline with 501 species don't occur in Brazil. Micromiene with more than 500 species and Winnertzine with 175 are represented by only 1 and 2 species in Brazil, respectively, while Cecidology, the most speciose subfamily, comprises 5,100 species. Among them, 265 occur in Brazil. Cecidology are divided into 4 subtribes: Brachyneuridae, Cecidology, Lasopteridae, and Stomatosematidae. 93% of the genera and 87% of the species are included in 2 subtribes, Cecidology and Lasopteridae. Why does it happen? Here is the explanation. These tribes comprise the gall-inducing species. In the world, there are 5,100 species of Cecidology, but only 500 occur in the neotropical region, corresponding to less than 10% of the global fauna. This percentage doesn't reflect the richness of the neotropical fauna. It only reveals the little knowledge of our fauna due to the scarcity of taxonomical studies. Brazil comprises about 50% of the neotropical fauna. This value is easily explained. Brazil has a very large territorial area with a very rich flora and is the most studied country. 45% of the genera in the neotropical region have never been reported in other regions, and until this moment, 46.5% are restricted to Brazil. Other amazing information is the number of species restricted to the neotropics, 93%, indicating that the neotropical fauna is very peculiar. Furthermore, 41% of the neotropical species are known only from Brazil. Brazil comprises 265 species of gall midges in 92 genera. Most are gall-inducers, but predators, fungivorous, inclines, and free-living phytophagous are also represented. Fungivorous comprehends 6 species: 3 primary fungivorous belonging to the basal subfamilies, and 3 secondary fungivorous belonging to Cecidology. Predators are represented by 12 species, and they have a distinctive larval morphology. Antennae are relatively long, body seta are long, and most species have pseudoptus for locomotion. Free-living phytophagous comprise 3 species of Cecidology that live mainly in flower birds, as Progyplosis floricola, a pest of potato, tomato, lime, and asparagus. Inclines comprise 9 species. An example is Stroteria quadridentata, whose larva develops in galls of Yangomia poulteria on Poulteria caemetum, a plant with edible fruits. Gall-inducers are represented by 210 species associated with 50 plant families. Fabaceae, Asteraceae, and Myrtaceae together host 37% of the richness of gall midges. These families are superhorse, with an average of gall-induced species by horse plant species varying from 1.6 to 2.5. Fabaceae, Asteraceae, and Myrtaceae are diverse in Brazil, being among the most specious angiospermas family in our country. This data adds evidence in favor of the plant richness hypothesis that predicts a positive correlation between gall insect richness and plant taxon size. When studying gall inducers, an interesting aspect to consider is the plant origin. In Brazil, 152 species of gall midges induce galls on native plants, 5 on exotic and 1 on naturalized. Concerning those associated with native plants, 50 species occur on endemic species to Brazil. Among them, 29 induce galls on endemic plants to the Atlantic forest, 4 to Cerrado, and 1 to Amazon. What does it mean? It means that these gall midges are co-endemic due to their species specificity. This is a very important information that should be considered in preservation programs. The number of endemic plants to the Atlantic forest and the Amazon is the same, about 8,000. Nevertheless, the Atlantic forest comprises the greatest number of co-endemic gall midges, simply because it is the most studied biome. In Brazil, Neolasioptera ramicola is the single Cecidology on a naturalized plant. This species induces galls on Physalis angulata, a medicinal plant known as joá de capote and used as anti-inflammatory. 5 species induce galls on cultivated exotic plants, which were introduced in Brazil. 4 gall inducers were introduced together, and surprisingly, the other, Phenodiplostis melissa, is known only from Brazil. How are the gall midges distribution in Brazil? First, let's see by phytogeographic domain. 72% of our fauna is concentrated in the Atlantic forest, 14% in the Cerrado, 7% in the Amazon, while the Caatinga, Pantanal, and Pampa together comprises only 6% of the Brazilian richness. In this slide, the territorial area and the plant richness of each biome are provided. We can notice that Amazon is the largest in territorial area and the richest in number of plant species. However, only 18 species of gall midges have been recorded in Amazon. The Atlantic forest is the third largest, the second richest in number of plant species, and the richest in number of gall midge species. Considering the floral richness, we can expect that Amazon comprises the greatest richness of gall midge species. However, this wasn't observed. In Brazil, insect gall inventories were carried out in almost all biomes, but mainly in the Atlantic forest and Cerrado. Nevertheless, the greatest gall richness was recorded in the Amazon by Julian et al. 2014. They found more than 1,000 gall morphotypes. Nevertheless, no gall inducing was identified in species. Other relevant information is that 70% of the gall midge species are known only from the type locality. In other words, the geographic distribution of most Brazilian species is poorly known. We can observe that there is a concentration of species in Rio de Janeiro, Minas Gerais, Bahia, São Paulo, and Espírito Santo, especially in Rio de Janeiro, and that there are several states with few or no records of Cecidology. Analyzing the host plants distribution, we notice that their area of occurrence is wider than that of the gum industries. This is true for all Brazilian hosts. This slide shows an example. The first map represents the distribution of the Alberja ecastophyllum, the horse plant, and the second, the distribution of Alopecia grandis, the gum industry. While the host plant occurs in several Brazilian states, from Pará to Rio Grande do Sul, the gall-inducing species is known only from Bahia and Rio de Janeiro. How to improve information on species distribution? Through new records, which can be obtained by collecting or herbarium visitation and examination of ecology execates based on the specificity of the gall morphology. Let's talk about Cecidology identification. In Brazil, in all gall inventories, most gall midges are identified in family. What explains this low number of identified species? I can list some reasons. First, immature specimens and adults of both sexes are necessary since many species were described based on different stages of the life cycle. Larvae are generally more easily obtained, as they are the longest lasting stage, being found in the galls, while adults are free-living and obtained by rearing. However, rearing faces several difficulties, such as fungal growth, dryness of the gall and of the plant, hypersensitivity, mortality caused by natural enemies, and unknown life cycle, especially the period of adult emergence. There are few little cycle studies on Brazil gall midges, and they were carried out mainly on species with induced galls on economically important plants, as Pisidium catriano, araçazeiro, and Eugenia uniflora pitangueira. Other reason is the great number of undescribed species and the low number of taxonomists. Phylogenetic studies on Brazilian gall midges are very rare. In fact, only 3 publications are known. The paper of Mon, 1961, on Asfondiline, but it was performed without using a cladistic methodology, and the papers of Maia Barbosa, 2018, and Garcia et al., 2020, both using a cladistic methodology to test the monophyly of 2 genera, Estephomeia and Brugmanniella, respectively, and the relationships among their species. Despite their ecological importance and the diversity of habitats and species, Cecidology are still little studied. Morphological, taxonomic, phylogenetic, biological, and distributional studies are essential to improve the current knowledge, as well as a new generation of taxonomists and, of course, employment for them. Thank you very much for your attention.
Now let's move on to the questions from Valeria in Dalva that the audience made. And the first one, let me see here, it's from Rosie for Dalva. Did the pests on eucalyptus enter Brazil due to failure in sanitary barriers?
I think so. Every year we have some pests coming here, coming everywhere. Like if you see in Australia and New Zealand that they have a very restricted barrier, they also get pests. And then here in Brazil, that our sanitary barrier is not too good, and also we have frontier with many countries. It's easy to introduce pests here. Okay.
Okay.
Could you please add Daniel, and then if he can complete some information?
Okay, of course.
Thank you.
Hello, I'm back.
Yeah. So let's move on to the next question for Valeria. Valeria, how many hours should I be in your lab to learn how to separate the Cecidology? So Valeria, I think your microphone, it's
Sorry. And now, can you listen me?
Yeah.
Yes.
Yeah.
Much better.
Okay. To separate, it's not. It isn't difficult to identify acetamide in family, but if you want to identify in species, the whole life. I am still learning.
Okay.
But I think that 2 weeks are enough to first know the morphology of the group.
So we have another question from Rosie to Dalva. Is there any attempted to control this pest in Brazil? Any successful case?
What pest do you mean? Any that may cause, any seal that may cause? Yeah. Okay, let's say one case, okay? We have Triazoida limbata that attacks guava, and guava is is very important cultivated in many states in Brazil. Here we use chemical control basically, but also cultural control, because guava is they cut every year that they can produce new growth, new flowers, new fruits. And then when you have new leaves, it's they are very attractive for seeds. And if you know the time the population of seed is higher, you try to do these cuts in different times that it's not, this population is not high. It's like, you see, cultural control or physical control. This is added to chemical, and we also have some natural enemies for this psyllidae. We have parasitoid and also predators. And in some cases, use the integrated pest management to control these. Okay, and then I think this is a case of success to control this pest.
Okay. So we have now a question to Valeria from René Carneiro. Valeria, you said that midges are a rich group worldwide. How has the speciation of the group occurred? Where's the center of diversification of the midges?
I don't know where is the center of the diversification of the midges. There's no studies about it. There are very local studies of radiation, but not in a global view.
Okay.
So I can't answer it.
So another question from René Carneiro, but this time is for Dalva. Dalva, are there papers that measure the extent of damage to the plants? Do you think this should be worth pursuing?
I think for some cultivated plants like Ilex paraguariense, erva mate, there is some papers measuring the damage of this insect. I think also on guava. But I think we can do this for other plants, but perhaps for other cultivated, like this tree for ornamental. Daniel?
I think in Europe there is, for instance, of the carrot psyllid. I think there are studies measure the attack or without attack on the effect on the roots and on the leaves. So there are these data for this. That's one of the things which comes to mind.
Okay. So the next question is for Valeria from Walter Araújo. Valeria, neotropical gall midges are most specialized by a higher proportion of monophytes than from other geographical regions?
Oh, that's a very good question. Carneiro et al., 19, I forgot the, no, 2029, measured the percentage of monophagous gall midges in Brazil. It was 92%. This value is higher than that I have seen in other countries, but I don't know if you can categorically affirm that. I think so. But more studies should be necessary because I don't know if in the Arctic, Afrotropical, Australia, there is a percentage of monophagous species. I should check to answer correctly, but I think so.
Okay. So Nicholas Kuzmanik asks for Dalva. Do you know any pest by Psyllid on Verbenacia?
Daniel, could you please answer?
I know psyllids on Verbenaceae, but I don't think they are pests. I seem to remember that I collected psyllids in Argentina on Verbenaceae, and there are some described species, but it's not a very common host family.
Okay.
Thank you.
So Christian Alfonso
Hi,
Villares
Christian.
asks for Valeria. Is it then possible to have a general biogeographical pattern regarding gall midges, or there is rather a large bias on collecting?
Yes, there is a large bias because Palearctic and the Arctic fauna are well studied, but Afrotropical, Neotropical, and even Australian fauna are still in study. We are beginning, so it's very premature to answer.
Okay. Elaine Cotrin Costa asks for Dalva, which of these pests cause the most economic losses in Brazil?
Let's see. Could be this Triazoida limbata, Daniel? I think so. Yeah,
So if we talk about the gall formers, I think it's probably Triazoida limbata.
Okay.
As this is wider distributed and occurs in many states, I assume it could be that. Otherwise, Gyropsilla spegazziniana is probably also important.
Okay.
But this concerns only the gall-forming species. The otherwise the most important pest economically is the Aphorina citri.
Yeah, if we talk about gall inducer, I think we can say it's Triozoida limbata, but in seeds in general is it's Aphorina. Thank you.
Rene Carneiro asks for Valeria, if you want to help increasing the records of gall midges, how should we collect and preserve the insects? What type of data is necessary?
Okay, well, if you find a goal already described with the inducer identified, I think that a photography or some samples of the goal are enough because they are specific in their morphology in the planting. So, but if you collect, oh sorry, ai. Wait a minute, socorro. But if collect a not unknown goal, it's necessary to obtain adults, larvae, and pupa in order these species be identified. So the material larva in alcohol in 70% alcohol, and the other material after hearing can be put in alcohol too. Alcohol 70%.
Okay, the last question we have here is from Marcelo Guerra to Dalva. What do you think about biological control of invasive plants using galling insects?
We have one example of a plant that was introduced in Florida. This is this Chinus terebintifoli, and they are testing a facility that make galls for control it. And then I think it's possible and it works. Perhaps Danielle can expand a little bit.
Yeah, in Florida there is another introduced plant from Australia that's Melaleuca quinquenervia, and this is controlled by a psyllid from Australia, Boreoglycaspis melaleuci, and there the success was really amazing. They could get rid of these plants. There is another example of a species from South America, from Argentina, which was used in Australia for controlling prosopis. And there it seems that it worked also. And the example of the Brazilian pepper tree in Florida, there was one species of psyllids which we described from Brazil, which was selected and tested, and it seemed to work very well, but it wasn't released because I think the cultures they had died more or less a week before it should have been released. So it will probably be released another time, but they have big hopes on that species as well.
Okay, we have a short time, but let's move on to the next presentations from Adriano and Gilson.
Okay. Yes.
Hi there.
Hello.
Shall we share my presentation now?
Yes.
Okay.
Okay.
Is everything okay there?
Yes.
Okay, good. Good afternoon, everyone. I would like to thank you, Jose and Bruno, for the invitation. And in the next 15 minutes, we are going to discuss some general information and some relevant findings about a poorly known group of insects called Thysanoptera, and some of them induces galls. And they are also called, commonly known as thrips. And the most distinguishing feature of these insects is their hairy or fringed wings, and they are very tiny, like the insects we have seen in the previous presentations, like psyllids and cecidomids. Most species have less than 4 millimeters long, and about 50% of the 6,000 thrip species known, they feed on plants, and around 1% of these species are known to be pests on several crops around the world. But there are also many species that feeds on fungi or are predators. But during this presentation, we are going to focus on the around 400 species of thrips that induces galls, like this one that occurs in Brazil. In contrast to most insects, galling insects, the galls induced by thrips are not induced by female oviposition. Perhaps one of the most famous thrips galls is that one that many of you know from ficus trees in everywhere in the world. They are simple leaf folds galls induced by a group of thrips in the genus Gynaecothrips, and most of the most gall-inducing thrips belongs to the family Phleothrippid, the most diverse family in Thysanoptera. And in this family, eggs are inserted in plant surface, so eggs are not inserted in plant tissues. So thrips galls are 99% of the times induced by feeding of the adults on plant tissues. So they have piercing and sucking mouth parts, and they drink the juice of the plants. But we still do not know how this process of gall induction starts in these insects. If the mechanical action, the mouth parts, which substance compounds are inserted in the plant, maybe through the saliva of these insects. They are all open questions. But we know that almost all of thrips galls are on leaves of several plant families, and there are a large diversity of shapes of these galls. But I'm pretty sure that the most common types around the world is that one, a typical rolled or curled, folded galls induced by thrips. That's what we usually see on the field. But also there are some shapes that we tend to say that are more complex, like rosette galls or true closed chambers that are observed in a very few cases in Thysanoptera. And you see that galls or thrips galls can be found everywhere, and like psyllids and cecidomids, like Valeria said, most species are known from Northern Hemisphere, especially on Indonesia, Australia, and we know very little about our neotropical fauna. But we had some nice advance in the last years. We had a lot of undescribed galling trip species around here, and during the last 15 years, we described 25 new galling trips from Brazil, especially on this genus, Holopotrips. And we finally got a name for several species that were on our shelves in the collection. So I think it's the hardest thing to do. It's to put names and identify the taxonomical diversity in such insects. And as for many group of galling insects here in Brazil, most species of galling trips are recorded from Mata Atlântica, from South and Southern Brazil, with very little known species from Northern region or Northeastern region of the country, where we have very important biomes like Amazonia, Cerrado, and Caatinga. We know very little about it. And it's purely problems with samples. So we do not have samples of that region. But when we finally got some indication about the identity and the biology in the field of these trips, we had very nice findings. And I would bring you some of these nice findings that we can get after knowing basic information about the taxonomy of this group. The first one is that the galling trips here in the neotropics, they like Myrtaceae plants. So if we list all the plant families that we usually find galling trip species, we'll see that almost all families, almost all galls are found in Myrtaceae plants. This is a pattern that we also found in other insects, but it's pretty strong here in trips. So the second more diverse plant family is Melastomataceae, with 3 species of trips inducing galls on these plants. So here we have some of the typical galls we usually to find here in neotropics, with the leaves of Myrtaceae, but we don't know why they choose Myrtaceae. Of course, it's a huge plant family, but I think there are more than only the number of these Myrtaceae species affecting this high diversity, and we are going to move forward this later. The second thing we discovered is that the gall-inducing behavior evolved at least 3 times in neotropical Thysanoptera. We did a revision of the main groups of galling trips in the neotropics, and we found that the galling behavior appeared in some very old lineages in the Phleotripid family, but also it's presenting 2 very divergent the diverse groups of trips: the Lyotrips lineage and the Holopotrips lineage. Interesting that this gall-inducing behavior was lost in this lineage several times in several different species. And especially on that huge lineage that is not shown here, but we have several trip species that lost their ability to induce galls, and they start to inhabiting and invading other trips galls. So they belong to the same genus. They are very close relatives, and we find two species living together at the same time in the same gall. So it's quite impressive that in some situations they still dependent on the shelter to reproduce and to feed. And the third one is that all the analysis we have been doing with plant, with gall morphology and gall anatomy, they say to us that trips galls are relatively simple in structure. So they are usually not enclosed chambers. That's why some of our colleagues do not consider trips galls as true galls and simply ignore them in the field. There is usually no new formation of tissues or specialized cells in these galls, and usually is restricted to hypertrophy of the cells and tissue hyperplasia. But actually, there are some curious changes in galls induced by trips here, especially on myrtaceous galls. They induce changes in the concentration of volatiles like terpenoids, monoterpenes, and sesquiterpenes, and these volatiles actually smells in the field. So several times when I was in the field, I actually smell trips galls. So it was easy to find sometimes galls using my nose. And when we went to the lab to see what was happening, we observed that in one species of myrtaceous galls, the tissue large amount of oil glands, much more than in non-galled leaves. And these oil glands that we can see here in OG, so these are the oil glands, are possibly responsible for releasing this strong scent in the field. So this is quite interesting. I never heard anything about a gall that actually smell, and actually is good. The smell is not bad. So what's the biological meaning of that? Maybe these scents are being released by the plant, or maybe these scents are being released by the trips. We don't know yet. Maybe the trips, the plants are using these scents in order to repel the presence of these gall inducers, or maybe, as in other situations, they are trying to attract natural enemies to kill the gall inducers, or maybe this scent is released by the trips, so they can attract co-specifics to mate, or maybe they are using it to repel natural enemies. So there are a lot of open questions, and there are a lot of space to research on that, on this these changes of chemical on the chemical profile of the tissues of galls induced by trips. In fact, there are much more galls induced by trips than we think. It's not only that very common gall we observed in the streets. Actually, there are lots of studies on plant anatomy and plant chemistry that will be very interesting to answer in the future. So that's what we expect doing in the future. Thank you for your attention.
Thank you very much, Adriano. Very good presentation. Now we're going to have Gilson's presentation. Welcome, Gilson.
Can you hear me?
Yes.
Okay, my presentation is on.
No, we don't see your presentation yet.
What about now?
Yes, now we do. But it's now full screen.
Okay. What about now?
Yes, now we do.
Okay. And what about the sound? The audio is okay?
It's okay too.
Okay. Well, good afternoon, everybody. On behalf of my students and collaborators, I would like to say that it's my honor to present our research, and I would like to say thanks to Rosie for the invitation. I'm going to talk about a small group of micromoths called the Cecidoses. Cecidosidae. As you can see here, they are very small, around 2 to 5 millimeters of wingspan. They induce galls in Anacardiaceae, in South America, particularly the genus Schinus, what we call Aruera. In Africa, genus Rus, also called the sumac plants. And the galls are typical, as you can see here on Eucecidosis minutanus. They are also called whistling moths because in our region, Rio Grande do Sul, with the wind, we call minuano, the galls supposedly whistling. And so the galls are called the whistling galls, and the plants the whistling plants. In Argentina and Uruguay, they call this mate or matecitos. They have an historical importance because the first species was described based upon material collected during the Beagle expedition, when Mr. Early, who was the assistant of Fitzroy, collected some galls and brought back to England, to Curtis, who described the first species that he called Cecidosis eremita. Until this century, practically we had only 2 papers following up that description. One of them, I am talking about measure papers, was put out by Bratton in Argentina, who for the first time formalized the family Cecidosidae, which means an allusion of Cecidium, a gall, and is the main family of Lepidoptera that induces galls. They are little known. as I said, and there is only one monography, a major one put out by a German researcher that came early in the last century to Brazil and published this monography on Cecidosis eremita. When we started work with them early this century, that what we had on hands. The taxonomy was, and it is, a little bit confused, as you see the reason by which we have that problem still to be solved. They were described as a tortricity. Later, Bratz created and proposed the new family Cecidosidae. But later on, they passed to be called Tinidae. And our colleague, a Brazilian researcher expert on Microlepidoptera, Vitor Becker, found their affinity with Incurvaridae and Prodoxidae and put them in Curvaroidae. And finally, we accept the Cecidosis family was accepted. There isn't a unique character of Cecidosis. How we identify them? As you can see, they are micromoths, small, contrary to the typical moth, macromoth. They don't have a proboscis. The gallia, the endite, is not developed, it's atrophied, although they have mandibles and other buccal pieces, apparatus, contrary to the macrolepidoptera. One particularity of the females a piercing ovipositor, as you can see here. The males valve have a pectinifer, kind of comb made of composed of little spines, and the wings have a modified frenulum that we called not a frenulum true one, but a modified one, a pseudo-frenulum. I don't want you to pay attention in detail about this slide. These are the characters that we had on mind up to early 21st century, where these authors, Howard Dugdale, proposed 8 apomorphy to the family. And none of them hold true today. The last one that is related to the monopoly of the genera that are valid is the absence of proboscis. Well, so this is what would put them in Curvarioidea, also associated with the ovipositor. But the absence of proboscis is something that could be a loss, since they are within the Glossata, although a very old family lineage. So this doesn't make sense and cannot be accepted a characteristic that would hold them together. So this character is not a good one, in my opinion. Most of them are, in fact, gall inducers, but we have one described in 2003 for New Zealand that is a leaf miner. So galls are not unique for the Cecidosidae. The gall-inducing has evolved several times in Lepidoptera. And also, another characteristic that you saw in that slide is that they don't have any attachment device ventrally, like legs or pseudopodium. This is not unique to Cecidosidae too. A lot of micromoth family have these characteristics. So what's left on is the Arnacardiacea. They are where in southern Brazil. It's as I said, a small group. We have now 80 species valid, in South America, 12 in Africa, and 1 in New Zealand. These are all associated with roofs, and the other 8 in South America with Schinus, particularly one species of Schinus, Schinus polygamous or Polygamus sensu-latus. Now it's split in several species, and Cecidology pampianus in Schinus vine manifolius, as I will show to you. You have already deduced that they have a Gondwanic distribution. At that time of Hoary Dugdale paper, with these diversity, as you can see on upper right on the screen, that was the distribution of the genera. Close to the end of last century, the group of Tillmeyer in United States worked with yucca moth, particularly the family Proboscis, proposed the first phylogeny for the family, placing the Cecidology close to the Prodoxis. They did a little bit work at that time with the markers that were available, and arrived to the conclusion that yes, they should have diversified circa 95 million years ago, and after the Lacardiaceae differentiation would split into two lineages, South American and African lineages, genera, that I will show to you in a couple of seconds. We worked out a little bit this proposition in 2017. A collaborator from Germany, Dr. May, was able to collect a few serots in Africa, and we came to this phylogeny. In fact, we have a proposed monophily for the family when compared to Prodoxidae, but the suspicion that we had that our genera that we were working on were close relatives to Africa, a little bit, I would say, did not fill our expectation. As you can see here, Dicranosis is the oldest one, and after we have the Cerotis from Africa, and all of the others, the most young lineages from South America. I will show you the biology in summary of all these species, at least one per genus. This is Cecidology eremita, as you can see, typical galls, the larva, the gall, it's an operculate gall. The mechanism by which these opercula are formed is not clear yet, but in fact, the pupa at the end of the larval stage and pupation opens the opercle, you have the adult. The adult will look for young branches, and with their piercing ovipositor lay their eggs. This is Eucecidosis minutanus, another whistling moth with typical galls, but much smaller, and that use the same plant, Schinus polygamous latus sensus. Here is the larvae, as you can see, the pupa and the adult. This is 1 millimeter. These folks are really small. This genus, Dicranosis, is a peculiar one and archaic. When it was first collected, those that did it, Dr. Biazanka, Southern Hemisphere entomologist, Russian one, in fact, sent to farms in the United States. He thought it would be a caddisfly and not a Lepidoptera, which is the sister group of Lepidoptera today. Because they have hairs instead of scales, they have larvae that don't have most of the structures that the other larvae have. And the galls erupted from the branch, close to the adult emergence, as you can see here. The other genus is Oliera, that don't have external galls. The galls are internal to the branch, as you can see here. They look like internal chambers that the pupa push close to the surface and protruded, and the emergence will occur. As you can see here, you'll find the exuvia, on the upper right corner. And this is the damage on lower picture. Old ones from more than 1 year, just craters of the Oliera galls. This is a new genus that was described recently from Rio Grande do Sul state. They're called Cecidonius, and the plant is a Schinus, different one. It's a very small plant, almost as herbaceous plant called Schinus vinemannifolius. The galls look like initially those of Oliera, the internal, but later on they are sterilized. And soon after being sterilized, they drop to the soil. They are dehiscent, and they will emerge. The adults will overwinter and pass to supposedly as a diapause state, or maybe quiescence, we don't know, and we will emerge as adult during early spring. Well, and finally, another genus that also has dehiscent galls was described recently by our group from Chile. As you can see here, central Chile, these are the galls, also in Schinus polygamous, the true supposedly Schinus polygamous. Well, the message that I would like to say to leave here, in order to attract future students to work with these galls, is first that they are little known, but there is a lot to be explored. For example, their distribution. This picture is the distribution of Eucecidosis minutanus. The arrows here are the 4 locations priorities study in 2018. Here, these red points are new locations coming from collections and herbarium data made by our group, and on green are the host plants. So it seems that something else, in addition to the distribution of the host plants themselves, determine the distribution. In other words, it's a little bit smaller in size, and in this case reduced to the Serra do Mar and the Cordillera de los Andes, the Andes Mountains. And we did a little bit more on this from a molecular perspective, and came up with at least 4 new lineages that speciate recently. And the genetic distance here is more than 3%. And we have morphological characters to recognize these species as valid. So only in this case, by doing this study, that we came to the conclusion that this species has a peripampasic distribution associated with old formation that probably with the uplift of the Andes lead to this peculiar distribution, an empty space in terms of presence in the Chaco region. And supposedly from this point of origin, progressively led to this speciation process, splitting this lineage on east and west of southern South America. Another study allowed us to conclude that they have crossed the Andes at least two genera, a new one that we called Cecidium, and the genus Oliera, are found on Skinus polygamous in the central region of Chile. Adding these species to the phylogeny that we had before, using African species, we came to the conclusion that Cecidiums is the newest lineage, with Cecidonius and Oliera. And this, as you can see, it's around 35 million years ago, so maybe before the uplift of the Andes. In other words, the Cecidonids were there before the uplift of the Andes Mountains, and that fauna is not unique to Chile because these species, Cecidium is a new one, and another one of Oliera have to be described. It's in our shelves to be described, but the message here is that the distribution is much broader than what we thought about the Cecidology. In other words, it's undersampling that's leading to this situation. Maybe they might go up to Peru and south to southern Patagonia. We don't know. We have to go after them. This is what I presented before. Is a kind of larger than expect, but we could go to the other end. They be very localized. That's the case of Cecidonius pampianus, where they have a distribution restricted to a region close to Porto Alegre City, where I am now. And as you can see here, each of these locations has its own haplotype, and they are organized by distance. In other words, each of these populations is isolated because they are located on small hills called the granite rills of Porto Alegre, surrounding Porto Alegre. And these species was described in the new genus under the threat of extinction because this is a savanna. The plants are herbaceous. They are considered as weeds by the farmers that for their livestock. And also we have other tracks like urbanization, cultivation of eucalyptus, pinus, etc. And so this is another aspect that I would like to leave here as a persuasive line of research for young students. It's not true that they all have external galls. As I showed before, for Oliera saiz in Chile, this is Oliera gertinana, that the galls are internal and never come to the surface. That's the reason by which this species was synonymized with Cecidoses 20 years ago, and we revalidated his status of valid species because we discovered that the galls were inside, as you can see here. Only the adults come out. So this is another aspect that our research should pay attention regarding these galls. This aspect of being dehiscent is also very important because you rarely will find these galls, as I show here, in the field. They fall to the soil, and what we will find here is only the scar of the gall, not the gall themselves. In fact, when you find the galls, you find them inside with maybe an attacked or dead larva killed by Apteromalaria or other microhymenoptera ectoparasitoids. As you can see here, a larva attached in the pupa. And instead of having the emergence of Cecidonids moths, you have a parasitoid. Or more interesting, the galls, as you can see here, are early modified by a second type of user that we call inquilins that modify these galls, from cylindric to large balls, as you can see on letter F, and on G, mature and dried in age. And that it's what called attention of the research and the public in general. That's the reason by which these galls were described as induced by cynipids in the old literature. And for more than 1 century, they were neglected by research. And this is just an example of what the difficulties that we have to work with these galls if we do not do intensive work. Just collecting them will lead frequently with taxonomic mistakes that are very common in the literature. Thank you all. That's the short talk, and I am willing to take your questions. Thank you.
Okay, thank you very much, Gilson. Now let's have the questions. Let's read the questions for Gilson and Adriano. So, first one is for Adriano from Elaine Cotrim Costa. Adriano, what phases of trips are necessary to describe a new species?
Wow. Usually in TRIPS taxonomy, we use adults only. So it's quite hard to match the identity of the larvae with the adults. They are quite different, and sometimes they are not in the same place. So it's quite hard to define a TRIPS species. So if we have information on larvae, good. Otherwise, we can work with adults only.
Okay. So Rosie Isaias asks for Gilson. Is the different mouth parts exclusive of galling micromorphs?
Could you repeat the question? Is what? Mouth part?
Is mouth parts, yeah.
No, they are not. They are not. They're very common among the Microlepidoptera in both stages, in larval and adult stage.
Okay, so Valeria Sidmaya asked for Adriano. Do only female trips induce galls?
That's a very nice question, Valeria. Actually, we don't know. We know that in several cases, one female foundress can start a gall, but in many times in the field, I usually see the early stage of galls with few females and one male and one female. The point is always the adult that starts the gall induction through feeding. But I think there's a tendency on trips that female foundress starts the gall induction. But it's a nice question.
So Bruno Garcia Ferreira asks for Gilson. How long may the cystidoses last inside the galls and out of the galls?
Oh, that's a really nice question. I would like just to complement Rosie's questions before, if you allow me. I forgot to tell you that they have shield mouthpart like other Lepidoptera in the larval stage. They feed upon the nutritious tissue that they are induced inside the galls, and Rosie is an expert on this in her group. And the adults, most of them do not feed as adults. Maybe ingest some liquid to hydration, but they are not active feeders. Most of them live hours or maybe a few days. Dr. San Blas and Don Davis in Argentina with gicanosis show that they die on the same day they emerge. They don't fly much. In other words, they are really, really local. Well, the next question is, Cecidology eremita has a 1-year life cycle, and I believe that most of them have that pattern in South and South America. Okay, there is a group that fly on October, November, another one on March, April, and in Chile is the same. So I would bet 1-year life cycle for most of them, but I don't have that information.
Okay. So Dalva Luiz Queiroz asks for Adriano. Is the species that attacks ornamental ficus an exotic species? If so, when was it introduced?
Good. Actually, this is the most common trips in the city. So those ficus trees came from Asia and were introduced in the cities in Brazil along the last century in mid 50s, 60s, and together with the trees, the trips came and they went very well here. And some of the people here in Brazil call them lacerdinhas because they, when they achieve a large population, they fly around and enter in the eyes of the people and scratch a lot. And people said in the past that it bothers like one of the major politicians in that time, Carlos Lacerda. So they call them Lacerdinha because of that, because they bother a lot in the eyes.
Okay. So we have another question from Rosi Isaias for Gilson. By the photos of the green galls coming from inside the stems, I guess their development do not involve stem bark. Is it true?
I think you are right, but I'm not the right person to ask that, to answer that. If you want to cut them, please just tell me that I will send you a few. But I think you are right. Yeah, I think gicanosis go deep into the branch. It has a different ontogenesis. Gallgenesis, is that correct? But it's just a feeling.
Okay.
Did I answer your question?
Yeah, I guess. True.
Yeah. In fact, another important information is that Eucecidos lay eggs into the young sprouts in shoots, and the other ones in older ones deeply into the tissue. They have strong ovipositors.
Okay. So Valéria Sidmaia asks for Adriano. Do Myrtaceae, the most gall plant family, only in Brazil?
Yes, I can. All our data say that. So if we go to other poorly known regions like Amazon, we probably will find other plant families, but I would say that Myrtacea there is also super host of trips.
Okay. So let's see. We have a question from Bruno Garcia Ferreira to Gilson. Are there many galls There are many gulls with unidentified Lepidoptera in Cerrado, rupestриan fields. Can they be Cecidocids or other Microlepidoptera?
I presume most of them are other microlepidoptera. We have been working in Cerrado recently with the collaboration of Dr. Alexandre Spechet, Planaltina. Have described a few and have many to describe. But up to now, the cecidoses have been restricted to Anacardiaceae, and they don't get into the Cerrado, most of the species. But some in the Atlantic forest, for example, Scinus angleri up to Minas and Rio, where I would bet someone could try to find them. Okay. And also in Lythria, it's not true that they are restricted to Scinus. We have found them in Lythria and other genera and other species of Scinus too. And the fact is that although we think that Scinus is well known, it's a poorly known family from a taxonomic perspective, a difficult one to work with. And there are even new species to be described. This last year, Dr. Borgion from our group put out a new species of Scinus from southern Brazil called Scinus pampianus. So it's a lot to be done. There is a lot to be done.
Okay. So Bruno Garcia Ferreira asks for Adriano. Which habits do the other lineages of thrips related to the three neotropical lineages of gall-inducers have? They are only herbivores?
Yes, interesting question. All those lineages that evolved the galling behavior are descendants of herbivores. So yes, it's quite difficult to observe the switch from fungivorous feeding behavior through herbivore feeding behavior in Thysanother. We observe the opposite. So we have plant feeders that start feeding on fungi, possibly on leaves, on the branches, and they start using only this kind of food. But yes, all the known trips, gall-inducing trips lineages are their closest relatives are also herbivores.
Okay. So René Carneiro, he commented, but it's not a question, but he commented, I agree that not only the plant family diversity is important, I guess they may radiate within the family by host shifting, but probably have more problems shifting to hosts of other families. And he made a question to Gilson as well. Gilson, do they all associate to stems? Is that a conservative characteristic across different species and genera?
Okay, thanks for the question, Rene. I would like to complement that I'm not saying that the Anacardiaceae are not in the Cerrado, okay? Is that I have never seen in Anacardium or other plants or other genera of Anacardiaceae cecidoses. No, they are not restricted to the stems. Most of Africa cecidoses are induced galls on leaves that is foliate and that fall to the ground and they jump. They're called jumping beans. A peculiar behavior. And we have two new genera here found on leaves, too, that to be described.
Okay, so Jack Schultz asks for Adriano. Are there any eusocial galling thrips species in Brazil, as there are in Australia?
Wow, that will be my dream to find some eusocial galling trips here as well. No, we don't have other social galling trips except in Australia. To the other people that are watching, we have a small lineage of galling Tysonatra that evolved an eusocial behavior. They are gall inducers in acacia trees, and the first generation develops as soldiers. They do not reproduce, and they have adaptations to attack intruders. They have very long spines on their forelegs, and they are usually very aggressive with other insects that try to invade. And thus, in the second generation, then came the reproductive insects that usually mate and disperse to other acacia trees.
Okay. So another question for Adriano. Dalva Luigi Queiroz asks, Adriano, working with psyllids in the field, we examined many galls from thrips, Diptera, and others. If we found galls from thrips, would you like to receive them?
Why not? It will be a pleasure to receive the samples, and it's quite interesting to work with good samples with many individuals, with the identification of the plant, so we can match the identity of the plant to the identity of the trips. And I think it's the same situation for all of the speakers said today. So this taxonomic information and this information from the field are quite essential to produce knowledge.
Okay. And Valeria Sidmaia asks for Adriano, do thrips gall smells only when induced in Myrtaceae?
Especially on Myrtaceae. Virtually all Myrtaceous plants with galls, they smell a very particular scent. But I also found this scent in other plant species like Piper, Piperaceae, and also Melastomataceae. So it's not found only on Myrtaceae, but mainly on such plants. So that's why I think that this scent is released by the insects, not by the plants. They are in some way collecting these oils or the plants and using it for a purpose that we don't know yet.
Okay. Marcelo Guerra asks, Adriano, did you compare the essential oil profile between galled and non-galled tissues?
Oh, yes. In one of the last study I showed, actually, the team, Rosie's team with Nina in UFMG, they worked in comparing the chemical profile of galled leaves and non-galled leaves of one species of Myrcia. And yes, we found increased concentration of sesquiterpene, especially on galled leaves, and a lot of volatiles on different volatiles in different concentrations on plants with thrips.
Okay. And Marcelo Guerra made another question for you. It appears that Tisanoptera like ferns too. I reported 6 records to ferns around the world. Did you find any thrips galls on ferns in Brazil?
No, this is not a usual situation. A usual thing we used to observe. There are some species that eventually feeds on ferns, but they are not specific. Actually, it's a very rare event for trips. We have few species on Central America that use ferns as host and produce some alterations. So if you have something, it will be nice to look at it.
Okay, so to finish, we have a comment and a question from Milton Mendonça. The comment is fantastic galling groups, both trips and micromoths. They add a lot of diversity in terms of the biology and ecology of the galls found around the world, around us. Thank you both speakers. And he made a question for Adriano too. Adriano, do you find parasitoid wasps attacking galling trips? If not, why do you think it is so?
Actually, the chamber that where trips lives, it protects them against the entry of parasitoid wasps. But we found in few situations some uh, uh, larvae of parasitoids that enter in the galls and eats on the trips. So the shelter provides protection against wasps, for example, by ovipositing inside the trips, but not protect them against the entrance of predators or natural enemies and the gall.
Okay, so thank you very much. We finished this session. Thank you, Gilson. Thank you, Adriano. Now we have a little break until the presentation of video posters with the theme Taxonomy and Diversity of Ground Users. Thank you very much. And see you.
Thank you, guys. See ya.
Thank you very much. See you tomorrow or later today.
Bye. I see you later.
Thank you for the attention. Bye.
Bye-bye.
Bye.
Bye-bye.
Hello, everyone. Welcome back. My name is Anna. I'm a biological sciences undergraduate student at the Federal University of Minas Gerais in Belo Horizonte. We will now start the video poster presentations, and they will be shown in groups. I'd like to call Ismael Cividini Flor from the National Museum of the Federal University of Rio de Janeiro, Brazil, Stella Esposito from Federal University of São Carlos in Brazil, and Universidade Federal Fluminense, also Brazil. Hello. If you have any questions, you can leave them on the chat and they will be answered after the presentations. All right, guys.
Hello.
Good afternoon, everyone.
Okay, so since Ismael is not here, I will present his video. So see you soon. Little problem here, sorry. Okay, since we're having problems with this video, Stella, can you present for us now?
Yes. Wait, just a minute. I will have to share my slides here.
Yes.
Okay.
Or you can present your video. Don't you have it?
Yes, I have it. Wait a minute. Hi everyone, I'm Stella and I will present you about my study, and the title is Spatial and Temporal Distribution of Galling Species in Cupaiferolangusdorff, a superhost plant. I did this work under supervision of Fernando Rodrigues da Silva and Maria Virginia Urso Guimarães. So first I want to introduce how our interest in this topic came about. Understanding the niche partition is necessary to understand how galling insects are distributed in superhost plants, which are those that harbor different galling species. Because they are species-specific insects, it is curious to understand how they share the niche. So, our main question is, how do galling species share the niche at the superhost plant Cupaiferolangusdorff? In this picture, we can explain the hypothesis and methods. The first hypothesis was spatial niche partition, and it means gall morphotypes will occur in different organs of the host plants. And two, gall morphotypes will occur in the same organ but at distinct position in the host plants. The second hypothesis was temporal niche partition. Some gall morphotypes will occur in the beginning and other in the end of rainy season. The third hypothesis was relationship local-regional. Regional richness is high while local richness is low, indicating high turnover of gall morphotypes among individuals of the superhost plant. So the method was because I went to the field and sampled 15 individuals of Kupffer-Langsdorff. In each plant, I observed the presence or not of new gall morphotypes. I want to share now our results. And the first one in this picture, we can see at the letter A, regional pool with all gall morphotypes recorded in the 14 individuals of C. Langsdorff. 14 because we did not find any gall morphotype in one tree. In letter B, we can see the number and composition of gall morphotypes by host plant. In letter C, we can see box plots showing pairwise dissimilarity. Here in this picture, in letter A, we can see the pairwise patterns of gall morphotypes since G1 to G19 of co-occurrence, showing probabilities that two morphotypes would co-occur at a frequency negative, positive, or random. In letter B, we can see distribution of the position in which gall morphotypes were recorded in the host plants, considering two axes: distance from gall morphotypes to the ground and distance from gall morphotypes to the tree trunk. Small circles represents all individual of gall morphotypes. In this picture, in letter A, we can see the temporal distribution of gall morphotypes sampled monthly from September 2019 to February 2020. In letter B, we can see principal coordinate analysis representing the variation of temporal dissimilarity. Now, our conclusions. Different from expected, we found no evidence that gall-inducing insects are inhabiting distinct organs in the same host plant, or that they are occurring in different vertical or horizontal strata. In contrast, gall-inducing insects showed temporal resource partition, with some species occurring from the beginning to the middle of the rain season, while others occurred from the middle to the end of the rainy season. We also found a low alpha diversity, with individuals of C. Langsdorff harboring different gall species composition. Now I want to thank CNPq, UFSCar, and my two supervisors, Fernando and Virginia. Thank you, everyone.
Okay, thank you, Stella.
Thank you. Sorry, the late.
No problem. Okay, now we will try this again. Okay, let's proceed. Bruno, can you present now? Okay, never mind. Let's try this again then.
Ismael Cividini Flor. I am a collaborating professor at the Museu Nacional do Rio de Janeiro. And the title of this work is Inset Gauss on Asteraceae in Brazil, Richness and Geographic Distribution. This work was done by me and my lab colleagues, Alene Ramos Rodrigues, Charlene Ascendino, Bárbara Proença, and Valéria Cid Maia. So, the family Asteraceae presents a large number of species registered in the row. In the last 30 years, several recent Gauss inventories in different Brazilian phytophysiomies have been published. Most of them comprise Gauss on Asteraceae and indicate this family as one of the most important in Gauss richness. The main goal of this study is to present a panoramic view of insect Gauss on Asteraceae in Brazil. The specific objectives to inventory the Gauss morphotypes on Asteraceae species in Brazil and to register the distribution of the Gauss in the phytogeographic domains. Insect Gauss papers in Brazilian areas published from 1980 until 2020 are analyzed in order to collect information about insect Gauss on Asteraceae in Web of Science between August and October 2020. We only consider the host plants identified at species level. Botanical names and synonyms were verified in Flora do Brasil 2020, as well as data on plant geographic distribution. Phytogeographic domains were retrieved from the original papers or verified in vegetation maps of Brazil. We found 88 insect Gauss papers in Brazilian areas, 51 of them with identified species of Asteraceae. This family is indicated as one of the superhosts in 21 inventories. The surveys totaled 487 insect Gauss morphotypes in association with 157 host plant species of 42 genera. This amount is higher than that compiled on Fabaceae and Myrtaceae by Santos Silva and Araújo 2020 and Maia 2018. Santos Silva and Araújo 2020 argued that Fabaceae are the principal superhosts of Gauss in Brazil. Our results pointed out that Asteraceae hosts a greater Gauss richness than Fabaceae. Asteraceae is the richest plant family in number of Gauss morphotypes in Brazil. The genera with the great Gauss species and Gauss morphotypes were Baccharis and Micania. Both of these genera are widely distributed throughout Brazil, and they are the best represented in the number of species with the Asteraceae family, favoring their association with the Gauss-inducing insects. The superhost plant species are Baccharis concina, Baccharis reticularia, Baccharis platypoda, Baccharis dracunculifolia, Baccharis retusa, Baccharis minutiflora, Elemantus erythropappus, and Micania glomerata. The presence of superhosts is common in semi-tropical regions, as contribution to the increase of the local and regional Gauss richness in the community. The Atlantic forest was the domain with the highest number of Gauss morphotypes, 200, followed by Cerrado with 150. 87 host plant species were recorded only in the Atlantic forest, 67 on the Cerrado. Most of the Brazilian insect Gauss inventories with Asteraceae were performed in the Atlantic forest and Cerrado biomes, which are relevant global diversity hotspots. The highest Gauss richness of Atlantic forest and Cerrado can be explained by the fact that these biomes have been more intensely surveyed, and additionally they house the first research center focused on Gauss insects in Brazil. One of the main conclusions that can be drawn from this work is that Asteraceae is the superhost family of Gauss in Brazil. The great majority of studies analyzing this work were performed in Atlantic forest and the Cerrado biomes. Finally, such studies are rare in Brazil and important to consolidate the current knowledge and to show the diversity and frequency of these insects in Brazil. Thank you for the attention.
All right. Now that that video is finally out of the way, Bruno, can you present for us?
Your microphone is off, Anna.
Shouldn't be.
Can you add the one? Pode, can you close the video?
Bruno, are you there?
Sorry, happen anything? Can I continue?
Yes, go ahead.
Okay.
Bruno, you have to unmute yourself.
There is no sound?
Try again.
Let me see.
I can hear you. Try your video now.
Okay.
Bruno, did you share your audio in the presentation?
I am Bruno Gomes da Silva from Universidade. Good afternoon. I am Bruno Gomes da Silva from Universidade Federal Fluminense, and I'm guided by Valéria Cid Maia from Museu Nacional UFRJ. This video poster is about the checklist of the gall midges in the state of Bahia. Galls represent discrete microhabitats that support relatively closed communities of specialist inhabitants. They are abnormal plant growths induced by various parasitic organisms, mainly insects, and provide the inducers with food and shelter at the expense of the host plant. Galls serve as incubators for the developing insects in which they gain nutrition and protection. Cecidomyidae is a very diverse family of Diptera, with more than 6,500 species. Most of them are gall inducers. The family comprises species of economic importance as an agricultural pest and other prey insects. In Brazil, only about 250 species of Cecidomyidae are known, most recorded in Atlantic forest areas of the state of Rio de Janeiro. In the beginning of the 20th century, a Portuguese priest, J.S. Tavares, described several species from Bahia. After this period, no new data was added to the fauna knowledge until 2014, when Maia reported 4 Cecidomyidae species in Porto Seguro and Trancoso. A literature review was performed on the database Web of Science using Cecidomyidae and Bahia as keywords. Brazilian inventories of insect galls and taxonomical papers were examined, as well as the most recent catalog of Cecidomyidae of the world. We used the Flora do Brasil 2020 website to obtain the data of host plants, and plant uses were verified in Santos et al. 2009 and on the Useful Tropical Plants 2014 website. 44 gall midge species of 28 genera have been recorded in Bahia. 39 are gall inducers. They are associated with 17 plant families, and among these, Fabaceae stands out for hosting the greatest number of gall midge species, 7, followed by Burseraceae, Calophyllaceae, Myrtaceae, and Nyctaginaceae, each one with 4 Cecidomyidae species. However, most plant families, 7, hosted a single gall midge species, and the number of host plant genera was not established due to 9 records in family level. Lopesia, Dazinura, and Brugmania were the best represented gall midge genera with 7, 5, and 3 species respectively. Most genera, 19, were represented by a single species, and 6 gall midge species induce galls in 4 endemic plants. 2 of them restrict to the Atlantic rainforest. 9 host plant species are useful, most with several uses. 6 are medicinal, 6 are used in carpentry and cabinet making, 5 in general construction, 5 are ornamental, 4 are edible, and there are many other uses. These useful plants hosted 19 gall midge species, 43% of the total. Most Cecidomyidae species, 39, have been recorded in the Atlantic rainforest, 11 in the Cerrado, and 7 in the Caatinga. Concerning the geographic distribution in Bahia, all Cecidomyidae species have been recorded in a single domain. Atlantic forest, 32 species, Cerrado, 5, and Caatinga, 3. 19 species are only known from Bahia. 18 of them were described between 1877 and 1922. And have never been recollected. Only one Cecidomyidae species, Schizomyia barreirensis, was recently described. 35 species were collected in coastal areas and 8 inland. Although the Atlantic forest is the phytogeographic domain with the smallest territorial area in Bahia, it comprehends most of the gall midge species recorded in the state. On the other side, the widest domain, Caatinga, comprises the lowest number of Cecidomyidae species. This result can be attributed to the greater number of studies of coastal fauna and also to the greater floral diversity of the Atlantic forest compared to the Cerrado and Caatinga. Regarding the categories of conservation, 12 plant species were not evaluated and none are endangered. Until the present moment, Bahia shares 19 Cecidomyidae species with Rio de Janeiro, 13 with Espírito Santo, and 10 with Minas Gerais. I want to acknowledge to CNPq for the productive scholarship and the audience present today.
Bruno, what happened?
Hello, can you hear me?
Yes. Did something happen?
What? Oh, I think it's fine.
Okay. We proceed to the questions now. Let me add Stella.
I'm
All
here.
right, guys. Great presentations. Now, I have one question from Jose. Hi, Stella. We have realized that some of C. Langsdorffii gall morphotypes vary in shape along development. Did you see something like that in your study area?
Yes, we have seen a lot of this because at first, when I went to the field, we collected all the gall morphotypes that we was finding, and we have collected first 25 morphotypes, and then we collected 19 morphotypes because some of them was only in different shapes of development.
All right. Another question for Stella. This one from Rene Carneiro. Stella, what do you think about the galls with positive relationships? Maybe I missed it. Are they on different plant parts or do they share the same developmental site? Don't they compete for food?
No, we found that they don't compete for food because there's a lot of available niche, and we saw that they happen by random. So we found this.
Okay. This one. I have a question. You tested if the gall's occurrence was related to each other. Why would you expect that?
Because our first question was that they compete for food. So we test the index of co-occurrence, and we saw that they happen by randomly. So that's why we expect that.
Okay. I don't see any questions. Oh, right here. From Jose. Bruno, did you compare your data with data of other Brazilian states?
Yes, we have compared with Rio de Janeiro, Minas Gerais, and Espírito Santo, and there's a few species with I think it's less than 20 species with common in Bahia and these other three states.
Okay. Thank you both for your presentations and for your time.
Thank you for the questions.
Thank you.
All right. Now I will call on Esau Ospina Penuela, sorry for my pronunciation, and Bárbara Proença do Nascimento. Esau is from Universidade Nacional de Colombia, and Bárbara is from the National Museum of the Federal University of Rio de Janeiro, Brazil. Hello, guys. Do you want to?
Hi. Hi, everyone.
Okay, Esau, can you start with your presentation?
Yeah, I think that Bruno shared the presentation for me. I don't know.
Okay, you can start.
This is Ospina, and I'm gonna present the work entitled A New Species of Britisemia maya, Diptera cecidomide, from Colombia with description of immature farms. Work done by me, Francisco Serna, Carlos Lamas, and Maria Virginia Urso Guimaraes. In this paper, we describe the second known species of the genus Britisemia from the Neotropical region, which is the first one from Colombia, as well as the first description of the larval form on the genus that was unknown. The genus Britisemia belongs to Schizomina, a subtribe with worldwide distribution, whose largest richness is found in the Neotropics. Up to now, the only representative of this subtribe in Colombia had been Schizomia manihori. Branches of the host plant were collected in a rainforest vegetation of the Andean Amazonian Piedmont in Colombia. The leaves with galls were placed into labeled plastic bags and transferred to the laboratory. Once there, some galls were kept in plastic bags until the emergence of the adults, and some were dissected to obtain the immature forms. All specimens obtained for reading were conserved in 80% alcohol for slime mounting. Schematic drawings were made using a drawing attachment connected to a light microscope. Digital pictures and electron microscope images were taken. Live specimens were deposited at the Entomological Museum UNAB of the National University of Colombia and the Museum of Zoology of the University of Sao Paulo. Britisemia resembles the genus Stephomia. These genera have neotropical distribution, and the host plants belong to the Myrtaceae family. Moreover, both of them have circumfilla reticulated palpus with one segment, female flagellomerous nitro 12 progressively shorter, mesobasal lobes present, and pupal antennal arcs short. However, Britisemia have other distinctive characters. For instance, in Britisemia, the ovipositor is longer than in Stephomia. Besides, the pupa of Stephomia has facial papillae, while the pupa of Britisemia has none. The adults and pupae from all collected specimens of the new species presented these distinctive characters. The adults of Britisemia colombiana are characterized by having flagellomere completely reticulate in the female antennae. Sternite 7 about twice longer than protusible portion of ovipositor, circus triangular and hypoprot weakly bilobed in males, unlike to Britisemia myrcia, who has circumfilla and completely reticulate, circus rainiform, and hypoproct deeply bilobed in males. The pupa of Britisemia colombiana has antennal arcs triangular, single-pointed, and about 4 times longer than in Britisemia myrcia, who has antennal arcs bifid. Third larval insert was described at the first time for the genus. It's characterized by one lateral papil situs on each side of the spatula and terminal segment without papillae, unlike Stephomia, which has 2 lateral papillae on each side of the spatula and 2 pairs of situs, terminal papillae. The larva of Britisemia colombiana has a spatula with 2 triangular and tapered apical teeth and ventral anus rounded. Both Britisemia colombiana and Britisemia myrcia induce lipped galls on species of Myrcia, a genus whose distribution is exclusively neotropical. Britisemia colombiana induce cylindrical galls and Britisemia myrcia tear-shaped galls. In conclusion, the genus of host plant associated with Bretaceaemia, has a wide distribution on the tropical region, particularly in South America, which suggests that the genus Bretaceaemia could be much more diverse and exist in different types of biomes in the Neotropical region than it was previously known.
Hi, my name is Bárbara Proença and I'm here to present my study entitled New State Record of Two Galmídeo Species in Brazil. This study aimed at report the first record of two galmídeo species to a Brazilian state. This work is part of the project Cisbiota Diptera that aimed at evaluate the diversity, systematic, and distribution of Diptera in 3 states of Brazil. One of them, Mato Grosso. This is the third largest state in Brazil. It is located in West Central area, and it is the only one with the presence of 3 physiogeographic domains, Amazon Rainforest, Pantanal, and Cerrado, but we only collected galls on Cerrado area. The galls were collected in Parque Nacional da Chapada dos Guimarães. This park is located in Chapada and Cuiabá municipalities. Its field work lasted 15 days, and they were done in September 2011, January and July 2012, and January 2013. All galls were photographed and transported to Laboratório de Diptera Neotropicais in Museu Nacional, and the host plants were identified by the botanics of Universidade Federal Rural da Amazônia. All the exudates are deposited in this institution. We separated part of the galls to obtain adults and another part to obtain the immatures. All the Cecidology were mounted in microscope slides. The species were identified based on host plant and gall morphotype identification, galmídeo morphology, and comparison with original descriptions. The insects are deposited in the Diptera collection of Museu Nacional. The first species collected was Anisodiplosis walteri. Galls of this species were collected in Velda Noiva Trail on leaves of Walteria indica malvaceae, a perennial herbaceous plant that is considered a common invader of cerrado areas and is found in all Brazilian territory, as we can see on the map at the right corner. The galls of Anisodiplosis walteri are spherical to conical, covered with short light yellow to light brown trichomes, and have one chamber where a single larva can be found. Anisodiplosis walteri was previously recorded in the states of Minas Gerais and Pernambuco, in areas of Atlantic Forest and Caatinga, respectively. This is the first record of Anisodiplosis in the state of Mato Grosso and in Cerrado areas. So in the map, you can see the current distribution of this genus in Brazil. The second species collected was Asphondilia sanctipetri, that induces galls on Didymopanax morototone and Araliacea. This plant occurs in almost all Brazilian states, as we can see on the map, and is very susceptible to fungi and insect attack. The galls induced by Asphondilia sanctipetri are green and glabrous and occur on leaf vein and petiole. We can see at the photo the details of the both galls. Letter B, a detail of the leaf vein gall, and letter C, a detail of the petiole gall. This galmídeos was previously recorded in the states of São Paulo, Minas Gerais, Goiás, in Cerrado areas, and Pernambuco in Atlantic Forest areas. This is the first record of this species in the state of Mato Grosso. In the map, you can see the current distribution of this species in Brazil. These new records are important because the Cicidomaia fauna is poorly known in Brazil. The gall midges have a distribution more restricted than their host plants. This could be explained by the scarcity of insect galls inventory in many areas of Brazil. So, according to the hypothesis of specificity of the gall midges, we assume that the both species may occur where the host plants are distributed.
Okay, guys, thank you for your presentations.
Thank
We
you so
have
much.
a few questions for you. First one from Jose. Esau, are there many entomologists working with galling insects in your country?
No, unfortunately, there are not many studies in taxonomy in this family. In my country, this is a large family, but is poorly known in Colombia. Just only 44 or 46 species had been reported or recorded in the country, and we began with this effort to improve the knowledge in and the work in taxonomy of this family.
I see. Another one from Jose, this one for Barbara. Barbara, by your experience, what is more common to find, new records or new species?
New species, because in my thesis I work with material from Rondônia and Chapada dos Guimarães, and I found 7 new species and only 3 new records, because most part of my my galls collected were new galls to science, but I didn't have the material of the insects to describe them. So I would say new species, for sure.
Okay, thank you. Another one for Esau, this one from René Carneiro. Esau, how many adults did you manage to get from the collected galls? How was the setup you used? I noticed you took the pupae out of the galls. Interesting.
Well, I collect more than 20 specimens, including parasitic of the family Braconidae and Eulophidae. Some genera that was recorded for parasitic Cecidomid, like Aprostocetus or but for it, I obtained more than 20 specimens. But when I try to reading in the laboratory, I have different issues, like mortality. It's very high, and reading problems, because I reading in the laboratory and the specimens have thermal shock and died.
All right. One more for Barbara. How do you explain such a disjunct distribution of uncidodiplopsis?
Mesodiploses. I believe that is combined facts like a few inventories. We need to inventory more because the plant occurs in all Brazilian states. Is a common invader. So maybe probably the Cecidomaia can occur in this plant in another states, not only Mato Grosso, Minas Gerais, and Pernambuco. So I believe that this is a problem done by the few inventories that we have in Brazil. Maybe if we inventory more, if we collect more, we will find this species in more states.
Yes, definitely. Thank you. One more for Esau. Awesome work in video presentation. What could be a reason this genus has only 2 species described currently?
Well, I think that currently the taxonomies around collection issues. One of them is that the research don't have a lot of or enough resources, economical or from the states or the countries to do our studies.
Okay, thank you. I don't see any more questions. So let me just put the others here. Congratulations, guys, for the excellent works presented here. Now let's call the organizing committee, which I only see one here. So, hi, Bruno.
Hi, thank you, Ana Maria. We solved the problems, and I hope that tomorrow everything is better than today, because even with these little problems which may occur in live transmissions, I think all the works were very rich. All the video posters, I want to thank you for the lecturers and to the lecturers and the video poster presenters for the amazing presentations, and someone more of the organizing committee is in the talking.
I think there's only you here.
Okay, so tomorrow we'll see together here in the channel of YouTube at 2 o'clock, 2 p.m. for 2 sessions and a session of video posters. The sessions are Goal Ecology and Interactions, and you have 3 lectures. And the session 3, Goals from ecological to molecular complexities. And the video posters, we have video posters on goal distribution and cool science. They are very interesting and very creative video posters. So see you tomorrow at 2 p.m. I hope everyone may participate how you participate today. Thank you and good night.
Good night. Good night, everyone.